additional research. Recording in progress. dance music. Research and Innovation Agency, a synergy in an orchestra. Established through the Presidential Regulation Number 78 of 2021 about the National Research and Innovation Agency. To enhance science and technology's contribution to the national development, and to improve the research and innovation ecosystem in Indonesia. The National Research and Innovation Agency is a government agency that is directly responsible to the President of the Republic of Indonesia. Consists of 7 deputies, supported by various research institutes, and have more than 70 research centers and supporting units. Through the 3 directions: science and technology resources mobilization, providing a favorable research ecosystem, building an economy foundations based on research. The presence of National Research and Innovation Agency is to achieve Indonesia's goals as a developed country in 2045. Also to make Indonesia as a global reference for sciences, bringing advantages on science and technology development, policy making, and societies. National Research and Innovation Agency. A synergy in an orchestra. Established through the Presidential Regulation Number 78 of 2021 about the National Research and Innovation Agency. To enhance science and technology's contribution to the national development and to improve the research and innovation ecosystem
Good morning,
in
ladies and
Indonesia.
gentlemen. I hope that you are well. So before we begin the event, I would like to read the conference guide. So the host, could you please try to share screen the conference guide? All right, so allow me to read the conference guideline. So for presenters and participants are expected to join at least 30 minutes before the conference is started, and all participants are expected to join all sessions in this conference. And please use the virtual background provided by the committee and rename your Zoom username according to the following formats. For presenter, you could use code of abstract or paper _name _institution. And for non-presenters, you could use the format P_name _institution. And please make sure to mute the microphone, especially during the presentation of keynote speakers, and in the parallel session, presenters should activate the camera. And questions from all participants are delivered using the raise hand, Q&A, or chat feature in Zoom. And when Q&A or chat feature selected, please deliver your questions according to the format name/institution /to name of the speaker and /questions. And every session will be led by a moderator. Those moderator will decide which questions is to be discussed or to be answered. And all participants should fill the list of attendees via the link given in each session and abide all the rules listed on the screen. So now we're waiting for Dr. Iman Hidayat to join us. Thank you.
National Re- National Research and Innovation Agency, a synergy in an orchestra. Established through the Presidential Regulation Number 78 of 2021 about the National Research and Innovation Agency. To enhance science and technology's contribution to the national development and to improve the research and innovation ecosystem in Indonesia. The National Research and Innovation Agency is a government agency that is directly responsible to the President of the Republic of Indonesia. Consists of 7 deputies, supported by various research institutes, and have more than 70 research centers and supporting units. Through the 3 directions: science and technology resources mobilization, providing a favorable research ecosystem, building an economy foundations based on research. The presence of National Research and Innovation Agency is to achieve Indonesia's goals as a developed country in 2045. Also to make Indonesia as a global reference for sciences, bringing advantages on science and technology development, policy making, and societies.
Ladies and gentlemen, may I have your attention? We would like to initiate the symposium in a few moments. The Honorable Dr. Laksana Trihandoko, Chairman of National Research and Innovation Agency, BRIN, Indonesia. Honorable Dr. Iman Hidayat, Chairman of Research Organization for Life Sciences, BRIN. Dr. Rati Asmananingrum, Head of Research Center for Biotechnology, BRIN. Head of Research Centers and Head of Bureaus in BRIN, distinguished speakers and moderators, representatives from ministries, institutions, and universities, and all distinguished participants. Good morning. It's a great pleasure for me to welcome every one of you to the 8th International Symposium of Innovative Bioproduction Indonesia on Biotechnology and Bioengineering, or ISIBIO 2021. Global Platform on Biodiversity and Biotechnology. ISIBIO 2021 is a continuation of the previous 7 seminars, first held in 2012, and has been annually carried out by the Research Center for Biotechnology, BRIN. This year, the symposium is held in collaboration with Indonesia Biotechnology Consortium, or IBC. The objective of ISIBIO 2021 is to provide a platform for academic dialogue on biodiversity and biotechnology, and for the exchange of recent updates on biodiversity and biotechnological researches. The International Symposium is conducted virtually live from Zoom meeting and official YouTube channel BRIN. Now, before we begin, allow me to read the conference guide. Presenters and participants are expected to join at least 30 minutes before the conference is started. All participants are expected to join all sessions in this conference. And please use the virtual background provided by the committee and rename your Zoom according to the format. For presenter, code of abstract or paper _name_institution. For non-presenters, you could use P_name_institution. And please make sure to mute the microphone, especially during the presentation of keynote speakers. And in parallel session, presenter should activate the camera. Questions from all participants are delivered using raise hand, Q&A, or chat feature in Zoom. And please deliver your questions according to the format, name/institution/2, name of the speaker, and /questions. And every session will be led by a moderator, and all participants should fill the list of attendees via the link and abide all the rules listed on the screen. Ladies and gentlemen, please be prepared and remain seated. We will sing to national anthem, Indonesia Raya. Ladies and gentlemen, we would like to begin the International Symposium with welcome remarks from the Chairman of Organizing Committee ISIBIO 2021. Please welcome Dr. ISIBIO.
Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. The Honorable Chairman of National Research and Innovation Agency, Dr. Laksana Trihandoko, the Honorable Chairman of Research Institution for Life Science, National Research and Innovation Agency, Dr. Iman Hidayat, the Honorable Director of Research Center for Biotechnology, National Research and Innovation Agency, Dr. Rati Asmananingrum, the Honorable President of Asian Federation on Biotechnology, Professor Dr. Duk J.O., the Honorable President of Indonesian Biotechnology Consortium, Dr. Mastur, all of our invited speakers from Indonesia, Malaysia, Japan, Saudi Arabia, Netherlands, and USA. Also, to our distinguished presenters, participants, and invited guests. Good morning, welcome, and thank you for being here with us today. Ladies and gentlemen, it is my pleasure to welcome all of you at the beginning of these two events. First, the 8th International Symposium of Innovative Bioproduction Indonesia, or ISIBIO 8 2021, and second, the 7th Indonesian Biotechnology Conference, or IBC 7 2021. Today, it marks our first event, ISIBIO 8 2021. It has been 8 joyful years that we host this annual event, and this year is special because ISIBIO 8 2021 is held in conjunction with IBC 7 2021. This would not be possible without a collaboration between Research Center for Biotechnology and Indonesian Biotechnology Consortium. So, in this opportunity, I would also like to acknowledge and appreciate the Indonesian Biotechnology Consortium and the team for making such a great collaboration. Distinguished guests, participants, ladies and gentlemen, this year, ISIBIO at 2021 has received high appreciation from 107 attendants who represent science, education, and industry communities, including 76 oral presenters, which 65 of them are paper presenters from various institutions and universities in Indonesia and Malaysia. As the output of this symposium, all accepted papers will be published in an international proceeding, IEP conference proceeding, and an international journal indexed by Scopus, Indonesian Journal of Biotechnology. On the third day of the event, IBC 7 2021 will provide you with two attractive talk shows. First, we will hear recent updates and the future of biotechnology startup companies in Indonesia from several CEOs of biotech startup companies, including Acumen Research Laboratories, Algae Park, and Nusantix. Secondly, the talk show will discuss program Merdeka Belajar, Kampus Merdeka, in order to share information to us about the opportunity of this national program in the field of biotechnology and biodiversity research. As the chairman of ISIBIO 8 2021, I would say, please make the best of this opportunity and enjoy casual yet meaningful scientific discussion with the experts on a wide range of topics. Just like the theme of this event, Global Platform on Biotechnology and Biodiversity, we expect that these events can act as a bridge for knowledge and technology sharing, and also an initial platform to spark research collaboration in the field of biotechnology and biodiversity. Last but not least, I would like to acknowledge all the sponsors who support ISIBIO 8 and ISIBIO 7, IBC 7. Tuwadia Prima Mulia, Elo Karsa Utama, Pandu Bioscience, Gitek Jaya, Nusantix, Merck, Crop Life, ITS Science Indonesia, Science Work, Gincraft, Biosense Medica, Berka Niaga Medica, and NutriLab Pratama. Thank you for the contribution and support for the events. We hope that these mutual partnerships will continue in the years ahead. To end my remarks, I once again would like to express my gratitude to all of you for your participation and contribution in this symposium. Thank you very much for your kind attention, and please enjoy the events. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you, Bapak Asep, for the warm welcome remarks. Ladies and gentlemen, next we will hear welcome remarks from the head of Research Center for Biotechnology BRIN. Please welcome Dr. Ratih Asmananingrum.
Thank you. Honorable Chairman of Organization for Life Sciences, National Research and Innovation Agency, Dr. Iman Hidayat, Head of Research Center of Organization for Life Sciences, National Research and Innovation Agency, President of Indonesian Consortium on Biotechnology, honorable speakers, honorable moderators, honorable invited guests, distinguished ladies and gentlemen. A very good morning to all of you. It is my great pleasure to address at the opening of the 8th International Symposium of Innovative Bioproduction Indonesia on Biotechnology and Bioengineering 2021 in conjunction with the 7th International Biotechnology Conference 2021. This year, the symposium and conference have the theme The Global Platform on Biodiversity and Biotechnology. The term biodiversity refers to the variety of life on Earth at all its levels, from genes to ecosystem, and can encompass the evolutionary, ecological, and cultural process that sustain life. Biodiversity is very crucial to our life for many reasons, from economic, ecological life support, recreation, cultural, to scientific. Any loss or deterioration in the condition of biodiversity can compromise all of the value and affect human well-being. Distinguished ladies and gentlemen, we are currently experiencing very serious problems related to climate change. It is one of the biggest challenges of our times. It is already happening. Temperatures are rising, drought and wildfire are starting to occur more frequently, rainfall patterns are shifting, glacier and snow are melting, global mean sea level is rising, land degradation is occurring, and biodiversity is decreasing rapidly. All the effects due to climate change have affected two important aspects of our life, food and health. Distinguished ladies and gentlemen, nearly 200 nations reached a climate agreement on COP26 to the role of fossil fuel in the climate crisis. The world needs to cut the rate of the greenhouse gas emission by almost 27 billion metric tons a year in order to limit global warming to 1.5 degrees Celsius by 2030. This effort certainly requires hard work globally and takes quite a long time. We certainly need a strategy on how to adapt and be ready to face climate change. Indonesia is the top 3 of mega biodiversity country, but the problem is that the use of biodiversity is still limited to provide solution to the nation problems, especially to encounter climate change. Distinguished ladies and gentlemen, biotechnology is technology that utilizes biological system, living organism, or part of these to develop or create different products. Research Center for Biotechnology, National Research and Innovation Agency is trying to continue to play an active role in utilizing biodiversity to answer the nation's problem. As a research center that has competence in health, food, and industrial biotechnology, we have carried out various research activities for food security and health. Several research activities for food security are development of stress-resistant and nutrient-rich transgenic rice and soybean, nutrient-rich and long shelf-life cassava, superior seeds cattle, probiotic-based feed for livestock health, and so on. In health security, we are discovering the active compound for medicine from microorganisms, plants, and animals. We also developing the biological-based product for infectious and degenerative disease. Distinguished ladies and gentlemen, this seminar and conference is held as an effort for all of us to share our competencies and experiences so that we can collaborate and work together to obtain output and outcome that can answer existing and future problems. We all hope that we can play an active role as part of the world to create a better life. Before I close my remarks, I would like to thank to the organizer committee for their hard work and valuable contribution to this seminar. And my deepest gratitude also goes to the Honorable Head of Organization for Life Sciences, National Research and Innovation Agency, honorable speakers and moderator, the sponsor for their great contribution, Indonesian Consortium on Biotechnology, and all of participants who attend this event. So as a representative of the organizer of the 8th ISIBIO and the 7th IBC, I officially open this event. Welcome to the symposium and conference. Please enjoy the scientific knowledge sharing, global platform on biodiversity and biotechnology. Thank you.
Thank you, Ibu Rati, for the wonderful remarks. Ladies and gentlemen, for the next, we will have a keynote speech by the Chairman of Research Organization for Life Sciences, BRIN, Dr. Iman Hidayat, and which is in this occasion representing the Chairman of BRIN. Good morning, Bapak Iman.
Good morning, moderator.
May I share
the presentation?
Before that, I would like to read a CV for Bapak Iman. Is that okay?
Yeah.
All right. So before we begin, allow me to read a short profile of Bapak Iman. Dr. Iman Hidayat earned his doctoral degree in plant pathology, Faculty of Agriculture, Chiang Mai University, Thailand. and he is currently the chairman of Research Organization for Life Sciences BRIN, and previously he was the director of Research Center for Biomaterial, Indonesian Institute of Sciences (LIPI). His research interests include development of advanced material technology based on plant lignocellulose and microbial chitin and cellulose. So without further ado, with Mr. Iman Hidayat, the time is yours.
Okay, thank you very much, moderator. I will try to share my PowerPoint first. Good morning, ladies and gentlemen, distinguished participant, keynote speaker, and all participants of the 8th ISIBIO and 7th IBC conference. I hope that all of you always in the good health in current condition and the pandemic COVID-19 condition, so that we cannot meet each other in this great opportunity. But we still can share our knowledge in the current conference. We can contribute to each other about our progress of research. Let me allow me to represent the Chairman of National Research and Innovation Agency BRIN, Pak Dr. Laksamana Trihandoko, to deliver policy on global platform on biodiversity and biotechnology research and innovation of BRIN. First of all, I would like to congratulate, of course, to the Research Center for Biotechnology BRIN for the ISIBIO 8 today, and also I would like to congratulate the Consortium of Biotechnology, Indonesian Consortium for Biotechnology, for the 7 IBC. In today's presentation, I will show several policy of BRIN in managing biodiversity, as well as in creating some strategy in biotechnology research and innovation to accelerate the contribution of research and innovation to the national economic development. We all know that Indonesia is a country with a huge biodiversity, from terrestrial to marine to freshwater biodiversity, from animal, plant, microorganism. This huge biodiversity is advantage or basic capital for Indonesia actually to develop economy that very important for Indonesia to develop the economy, because the biodiversity, at least it has economical value, it also ecological value, and also it has cultural value. So it is important for us as a researcher and academic to conserve as well as to utilize the biodiversity. However, in managing biodiversity is not easy. Many countries with huge biodiversity belong to developing or poor countries. I don't know why, but because the managing biodiversity is not easy. We, how can I say? We have to maintain, we have to unreveal the biodiversity, we have to develop the technology to utilize In the same time, the growth of human population also become problematic for, especially for Indonesia, to manage the biodiversity. Growing of population, it means growing in food demand. It means it will need more area for them. It means growing for industrialization. At the same time, the world climate change also increasing. It is, how can I say, it's not easy to managing biodiversity. So in managing biodiversity, at least there are two parts. The first one is how to conserve the biodiversity for future, and then how to utilize the biodiversity for economical development. BRIN has the responsibility in Indonesia to carry out both function, conservation and utilization. For conservation, it is the responsibility of BRIN to provide ex situ conservation. In the ex situ conservation, BRIN provides the biodiversity exploration and inventory program, human resources, as well as supporting funds. And BRIN also provide the facility for the national depository of plant, animal, and microorganisms. And also BRIN responsible for all stakeholders in Indonesia, not only Indonesia, in other countries as well, to access all the biological collection and information preserved in culture collection, herbarium, and museum managed by BRIN. And BRIN also provides services for the identification of biological diversity. For the plant conservation, ex situ plant conservation, BRIN also developed National Botanical Garden. Until now, there are 4 National Botanical Garden, Bogor Botanical Garden, Cibodas Botanical Garden, Purwodadi Botanical Garden, and Bedugul Bali Botanical Garden. Next year, BRIN will develop more national botanical garden, at least 5 more national botanical garden. And in the same time, BRIN collaborate with local government to develop local botanical garden. It is important because with the accelerating of industrialization and also climate change, global climate change, many plants is now going to extinct. It is the responsibility of BRIN how to conserve the plant before extinct. In the utilization, there are also several examples of BRIN to develop a low-level technology and also high-level technology. So back to the biotechnology, there are many definitions, but one of the simple definition is the manipulation of living organisms or their component to produce useful product. So biotechnology is the key for the economical development based on biodiversity resources. It can be for bioenergy, medicine, veterinary, agriculture, fine chemical products, and pharmaceutical. It is challenge for academicians and researchers in Indonesia how we can deliver all of these challenges. Where are we now? We know each other. Where are we now? Do we have this product until now or not? It is the challenge for us. So the current condition We know that biodiversity is theoretically the capital for economical development. And it is the great advantage for Indonesia. And the tool to make it is biotechnology. Unfortunately, until now, we still left behind to other countries, and we has not taken full advantage of this potential. We conduct several biotechnological research and several products such as superior variety of staple food, paddy rice, cassava, and also we do research how to develop superior livestock from local varieties, and also we develop vaccine now, as well as how to develop, we develop various vaccine, not only SARS-CoV. But the challenge is not only that, because this biotechnology is the key for food security and health resilience. As Dr. Rati previously mentioned, biotechnology is the key to nation food security and health resilience. But we witness now, until now, that Indonesian national health resilience is still weak and still not meet the demand. I can show you the case of study for the COVID-19 case. We know that the first case of COVID-19 is in November 2019 in Hubei. The first genome or SARS-CoV was announced in January 2020, and Indonesian first case of COVID in March 2020. Since the first cases, around 6 months from the first cases in Hubei, Sinovac already conducted phase 1 and 2 human clinical trial. And they get the result in June 2020. And Indonesia first administered of COVID-19 vaccine was conducted in January 2021. Until now, already about 46% of Indonesian population has received vaccine of COVID-19. Compare to the Chinese vaccine or Chinese biotechnological research, Merah Putih vaccine have just finished preclinical trial on macaque last month. So we know that there is huge gap between China biotechnological and Indonesian biotechnological. I will show you several bottleneck or constraint in the development of vaksin merah putih, in Indonesia. So there are several technology to develop vaccine inactivated virus from the viral vector and so on. But the bottleneck is Indonesian researchers and academicians had never developed vaccine. So in terms of experience, skill, we are left behind. The second one, collaboration among researchers, academicians, and industry is also need to be improved, something like that. And the last one is infrastructure. To develop vaccine in Indonesia, we don't have GMP facility. We don't have certificated animal BSL-3 facility. So it become a great problem for us in the pandemic, how to develop the vaccine. To answer this problem, so BRIN start to develop infrastructure and then how to develop human resources as well as how to develop the research and innovation environment in Indonesia. Now BRIN developed several facilities in Indonesia, not only in Cibinong, to support the research and technology to utilize biodiversity in Indonesia. This is the building of Indonesian Biodiversity Collection for and data center, and the building will be operated in August next year. And this is the Genomic Biodiversity and Environmental Laboratory building to conduct research on genomics, protein engineering, cell development, cell manipulation, and so on. It will be operated in January 2022. And also BRIN also have BSL-3 facility to develop infectious research, disease research. And also BRIN also build the integrated laboratory of bioproduct in Cibinong. All of these facilities are in Cibinong. This not and BRIN also build some facility not only in Cibinong but also in other region in Indonesia, including in West Java, in Sumatra, in Ambon, in Lombok, West Nusa Tenggara, and so on. All are open access for all stakeholders. So not only researcher for BRIN, not only researcher from BRIN that can access all the facility, but other stakeholders, academicians, industry also have the opportunity to access and to utilize the infrastructure through ELSA system. So BRIN developed the system so that all stakeholders can access and can use the facilities. In terms of researcher and industry relationship, BRIN also developed the platform how industry can get involved in developing products. Until now, in Indonesia at least, researcher or research institute in Indonesia doing their research without involving industry. So at the end, mostly the product do not meet the industrial standard or demand. It become chronic problem in Indonesia. Therefore, BRIN now start to involve the industry from the research planning until how can I say, from the planning until the execution, the research involve the industrial resources. So BRIN in this context as a multi-stakeholder collaboration hub, it is not only involving industrial stakeholder but also other stakeholders. To develop human resources, BRIN provides several platforms. For example, visiting researcher, assistant researcher, research assistant, and visiting scholar. It is important not only for BRIN, but also for other stakeholders to develop the mutual relationship. For example, in the research assistant, student that conducted research at BRIN has the opportunity to become research assistant and then has the opportunity to continue their study full funded by BRIN through by research program. So it is a great opportunity for students in Indonesia as well as for academicians in Indonesia. Not only that, but also to other stakeholders, so they can increase their competency, knowledge through the system. So BRIN will facilitate research partnership, stimulate multi-stakeholder collaboration, and stimulate the innopreneurship. And we hope that for the next 3 or 5 years, we can harvest the output from this program. I think that's the presentation that I can deliver regarding the policy of BRIN to accelerate biotechnology, research and innovation in Indonesia. Thank you very much.
Thank you, Bapak Iman, for the very insightful speech. Ladies and gentlemen, before continuing to the next agenda, we will have a photo session with all participants. So could you please kindly open your video and the host will help us to take a picture. All right, so there are 7 slides. We will start with the first slides. For the photo, could you please try to give your best smile in this fine morning? Okay, so we'll start for the first slide. 1, 2, 3. The second slide. 1, 2, 3. The third slide. 1, 2, 3. The fourth slide. 1, 2, 3. The 5th slide. 1, 2, 3. The 6th slide. 1, 2, 3. And the last slide. 1, 2, 3. Thank you, ladies and gentlemen, for your participation. Distinguished guests, ladies and gentlemen, we will continue to our next agenda, which is plenary session with speakers Professor Junichi Tanaka from Department of Chemistry, Biology, and Marine Science, University of Ryukyu, Japan, and Dr. Mastur, President of Indonesian Biotechnology Consortium, or KBI. This session will be moderated by Professor Ekowati Hasana. And before we begin, allow me to read a brief profile of Professor Ekowati. Professor Ekowati Hasana achieved a doctoral degree in Food Science Program, Biotechnology, IPB University, and master's degree in Department of Food Science, Nutrition, University of Rhode Island, and for Bachelor, Agriculture Technology, Universitas Gadjah Mada. She is currently a professor in Research Center for Marine and Fishery Product Processing and Biotechnology, Ministry of Marine and Fisheries, Indonesia. Professor Ekowati has been conducting researches in the area of marine biotechnology and fisheries product. Now, without further ado, Professor Ekowati, time is yours to lead the session. Good morning, Professor Ekowati.
Okay, yeah. Now, ready. Thank you, Mbak Wiwit. Assalamualaikum warahmatullahi wabarakatuh. Honorable speakers, distinguished guests, and all participants, good morning. Welcome to the plenary session of the 8th ISIBIO 2021, and in conjunction with the 7th Indonesian Biotechnology Conference, IBC. In the next 1.5 hours, I'll be the host of this session, serving as your moderator. Today, we will be hearing presentation from our speakers, Professor Junichi Tanaka from Japan and Bapak Insinyur Mastur MSc PhD from Indonesia on this very timely subject. But before we get started, please allow me to introduce our speakers. Professor Junichi Tanaka is a professor at the Department of Chemistry, Biology, and Marine Science, University of Ryukyu, Japan. He has been attached with the university since 1996 as instructor and as associate professor in 2007, and finally appointed as professor in the University of Ryukyu since April 2007. He is also alumni of the University of Ryukyu for his master and his bachelor degree. And he achieved his doctoral degree from Pharmaceutical Science, Osaka University. His research interest is on new bioactive compounds from coral reef life, particularly invertebrates. He also engaged in collaborative research with other researchers on related research such as mode of action, structure, activity, relationship, biodiversity, and biosynthetic genes. He has a lot of experience working abroad as a visiting scientist. In 1991, he was visiting scientist at Chulalongkorn University, Thailand, and in 1993 and 1995 at Parmamar, Spain, and in 1997 he was with LIPI Oceanology Indonesia, and in 1998 2007, he was with Max Planck Institute for Biochemistry and Saarland University in Germany in 1998 and 2007. Now, please welcome Professor Junichi Tanaka, who will be present marine natural products and biodiversity of marine organisms. Prof. Tanaka, are you there?
Yes, thank you.
Yes. Your time will be 40 minutes, Professor. Okay, time is yours now.
Okay. Thank you very much for the kind introduction, Professor Ecology, Hassana. And okay, let me upload my slide. Okay. Do you hear me? And also, do you can you look at my slide?
Yes.
Okay.
Yes.
Okay. Let me start. Okay. I'd like to thank the organizing committee for inviting me as a plenary lecturer today, and also my former student, Dr. Penny Amadi, for arranging this opportunity. It's a great honor for me. Okay, before talking on the contents, let me introduce where I am based on. if you compare Okinawa from Indonesia, it's so small. Okinawa is located at southwest part of Japan and it consists around only 60 islands or so. And it is located east of Taiwan.
Sorry, Prof. Tanaka.
Any problem?
Could you make your voice loud? Could you make your voice louder?
Okay, I got
Louder, please.
Is it okay now?
Yes. Yeah, it's better.
Okay, sorry. My voice is usually small. Anyhow, let me continue my talk. Okay. Anyhow, I am from this island, and the island of Okinawa is around. It spans around 500 kilometers in distance. Anyhow, thanks to the warm glacial current in the Pacific, the islands are washed by warm water. So, just like as in Indonesia, the islands are surrounded by coral reefs. Our university is located on one of the island, largest island, and these are our buildings. And anyhow, it is located at a nice place to work on chemical and biological aspects of coral reef organisms. Okay. Today, I like to talk about marine nature product and some of the diversity work we do. But after the brief introduction on marine nature product chemistry, I'd like to talk about some typical natural product studies, including an example of structural illustration or determination. Some of you may be not familiar with chemical analysis, so please. But it's just part of the basic matter for our field. And we usually look for newer molecules from the marine sources, but even a normal natural product, there is some application. I'd like to show one example. And during natural product study, sometimes you may need to be careful on the research. And I'd like to show one cautious example. As a second topic, I'd like to show some bioresources in nature and also a phenomena of some marine organisms. And so I'd like to show some useful or precious molecules from marine organisms, and also some unique phenomena of metabolites and organisms. And at the last, I'd like to talk about chemical and genetic diversity of marine organisms, of two groups of organisms. These are Cyphyton, Sophorus, and Iridia mutans. Okay. All right. Natural products are, of course, metabolites produced by organisms. Plants have been utilized from ancient times, but microbiomes metabolites may be utilized from middle 20th century. Compared from then, marine sources, marine organisms are relatively new, maybe utilized after maybe after 1970. One of the pioneers is late Professor Shoyu of University of Hawaii, and he was dubbed as the father of marine natural products. And why are we attracted by marine organisms? The answers may be the huge number of species in the water. Once it was expected that more than 500,000 species, but some paper reported this estimate, but every year the number is increasing. So I'm not sure the exact number of species. Anyhow, there is a very huge number of organisms in the water. Not only sessile marine organisms, but there are also marine microorganisms. And of course, we can expect the environment is totally different from terrestrial area. Apparently, it is aquatic environment. They operate with salinity and pressure, etc. So, after the long evolutionary history of new organisms, they develop different strategies from that of terrestrial organisms. And also there are a number of relationships between organisms. Some are probably parasitic, symbiotic, etc. So, yeah, probably it's endless target. Okay, let me talk about some basics of my method for study with our new example. Okay. First is typical structure determination work, because we are dealing with chemicals, so without knowing what kind of molecules we have, there we cannot proceed further. Okay. So currently, a group of compounds originally reported by Australian workers. And when we worked on a cytotoxic constituent of this sponge in Okinawa, my student isolated two new molecules. These are stereoisomers, or diastereomers to the known ones. One of them, named as Franklin X, is a non-crystalline molecule, but after many trials, my student found that its derivative, having bio in the structure, can be crystallized. And by using X-ray crystallography with diffractometer, you can reduce the whole stereochemistry. With X-ray diffraction study, usually nobody claims that your conclusion is wrong or no. But when you do crystallography, it totally depends on the nature of the molecule, whether it is crystalline or not. In case of, as I told, in case of Franklin X, the original material was not crystalline. And according to Professor Fujita of University of Tokyo in Japan, it is a 100-year-old problem whether the molecule is crystalline or not. Even now, we cannot predict which molecule will crystallize or not. But he invented a so-called crystalline sponge nest to solve non-crystalline molecule by crystallography. I heard that currently it can be applied only to small molecules, but hopefully his group invents a molecular scaffold to solve various molecules in future. Yeah, it is developing. Okay, another molecule named Franklin Y was isolated together with Franklin X in the previous slide. In this case, we could not find any crystalline derivatives. So we have to solve the stereochemistry with NMR instrument as well as most conventional natural product studies. But in this case, although it seems to be clear on the spectrum, but there are some signal overlaps, and we face a little bit difficulty on assigning the stereochemistry. So once again, my student try to make several derivatives, and one of them having alcohol at this position instead of double bond. And after doing so-called NOE study and finding out the stereochemistry. Those are the typical works. But in another case, it was not so easy. When we worked on a cultured microalgal metabolites for antiviral screening, we isolated a new molecule named caveolin in relation to a known molecule, protein. Protein was already reported as a site constituent, but we also reported its crystal structure. But in case of caveolin, not like protein, it was not crystalline. So we used the NMR to illustrate the whole stereochemistry. But with NMR study and additional works, we can only find that 5 partial stereochemical structure colored in differently. So the final molecule must be one of the possible structures. So in this case, I asked my colleague, calculation chemist, to estimate optical rotation value to compare with the actual observed value. So my colleague generated 16 possible structures, and after calculation, he found stable conformals. Then he calculated optical rotation value. And in our final estimate, the whole stereochemistry may be like this, but it is just estimate. In future, I hope synthesis chemists may conclude whether our tentative structure is correct or not. Okay, second topic is the application of a non-natural problem. Polychondramide is a member of trisoxazole macrolide, having 3 consecutive oxazole moiety in the macrolide. This member of molecule was discovered only from marine sources. And they are graphylide, caveolamide, and polychondramide, and microlide, and their congeners. And their mode of action or cytotoxicity or antifungal activity were caused by targeting site actin, the most abundant protein in our body. And with my collaborators, we reported how the macrolide bind to the target protein actin, and also we could solve the whole stereochemistry by crystallography with protein. And we also understood the mode of action. Okay, also the structure is quite complex. We can obtain the pure compound of this molecule from natural sources in gram quantity. And this situation is quite different from some other members, such as capillamide, which are quite complex as a as a as a mixture of similar molecules. So we use for some other purpose. In addition, the molecule contains enol moiety, which is a good target for our nucleophilic reagent. So we made several ellipses based on these molecules. At first, we prepared simple derivative named the ISIBIO A and its ephemer by treating icondiamide with ammonia water. By evaluating cytotoxicity against cells, culture cells, and also affinity to fluorescent GRP, it seems to be that the ISIBIO and ephemer has a higher affinity than original icondiamide. Second application was to make a fluorescent probe. As we know that this class of molecule binds to the leading edge of actin fibers in cells, we plan to stain the peripheral region of cells. By attaching fluorescent moiety through shorter or longer linker moiety, the sustained cells, like as shown, as in the right. Okay. The last application was making affinity gel. Nowadays, molecular biology method is used for finding the amino acid sequence of proteins, but I wonder if there might be an opportunity to isolate actin-like proteins. In this case, affinity gel with shorter linker did not work, but with a longer linker moiety, the gel is likely to work. Okay, in the third topic on natural product, I'd like to discuss about artifact formation. When you isolate molecules from natural sources, you may believe that they are natural, but it's not. There are quite a number of possibilities to isolate non-natural molecules, namely artifacts. Here I listed causes to make artifacts. 3 example molecules are shown. The first one is acetone adduct of aldol-like reaction. Second one is hydroquinone, because as you know, it's easy to be oxidized to quinone. And the last one is contains a lactone moiety, but under mild acidic condition, it cleaves into carboxylic acid and alcohol. Okay. So it totally depends on the structure and the conditions when you handle these molecules. In the papers published by my neighbor laboratory, working on my models as well as ours, they reported some molecules with hydroperoxide moiety. These are hydroperoxide. As a hydroperoxide signal in NMR is conspicuous, appearing chemical shift 7 to 7.5. So we believe that we did not look over it. However, we have never encountered molecules with this functionality. When we think about the difference between the 2 laboratory conditions, there is a significant difference. Our neighbor laboratory has a big window facing south. That means we are located in the northern hemisphere. Sunlight penetrates through window, and they don't use window shade. In our laboratory, our windows are tree shaded for the south part, and the windows are facing north. So we examine the possibility of sunlight. It is already known that singlet oxygen will be formed from normal triplet oxygen, which we breathe, not directly with the light, but indirectly through excited photosynthesis fiber or pigment. As most of extracts from natural sources contain pigments, so it might mean that singlet oxygen may be formed if solution of the material is exposed to sunlight. And in addition, it is a well-known phenomenon that singlet oxygen will react with a molecule containing double bond to give hydroperoxide through in reaction, as shown in this scheme. Compound 1 is a stable marine natural product from a sponge, and we kept it in a freezer for 3 decades and use it as a model molecule. We incubated in compound 1 with artificial pigment named rose bengal, with or without sunlight. And of course, without sunlight, there's nothing happens. But as it is expected, and if it is exposed to sunlight, hydroperoxide, compound 2 was formed. But when we left the mixture for 2 weeks by the window, it resulted in the isolation of further oxidized or degraded molecules 3 to 5. Particularly compound 5 is highly oxidized to incorporate the peroxide in the molecule, which is a little bit similar to the known anti-malarial drug artemisinin. With conventional oxidizing agent, I believe it is difficult to oxidize position like here. So it might be useful to make some unusual derivative, though the reaction speed is very slow. Okay, the formation of compound 2 and 3 can be explained as in this scheme. The air reaction product 2 will react with water, namely Fock reaction, to give hemichemical compound 6. This compound will flip to give compound 3 tetraldehyde. But for other products 4 and 5, it is not easy to explain how they are formed. After publishing our results early this year, we noticed that enantiomers of compound 1, 2, and 5 were already reported by high researchers as sponge metabolites. On the other hand, Israel group reported another similar molecule named axiprins as sponge metabolite. I don't know their laboratory conditions, but we have to be careful when you handle the material under sunlight. Okay, I summarize the first part of my talk. When you work on structures of natural products, suitable methods should be chosen for determination or elucidation. We usually neglect or ignore non-natural products, but even non-molecules can be used for some purpose. And because, as I thought, it is better to avoid sunlight to avoid artifact formation. But if you want to have some unusual oxidizing metabolite, singlet oxygen might be useful. Right. For the second part of my talk, Now, in this section, first I'd like to show a few cases of bioactive metabolites from and their sources from Indonesian water. In the latter part, example of the same or similar compound from different organisms are shown. As I worked on some marine resources with my collaborator in Indonesia and Japan, I'd like to show some resource organisms. Jaspamide or just platinolite, one compound has two names because they were different. They were reported by two groups independently. Anyhow, this kind of molecule is a DEPS peptide, so-called DEPS peptide, and showing active polymerization activity. And it is sold as a life science reagent with a good price. This source sponge spread from the Philippines to Indonesia, and you may supply the molecule, though isolation is a little bit difficult. Zesta sponge C is a unique alkaloid molecule and sold as a reagent as an IP3 receptor antagonist. This sponge distributes from Okinawa to Indonesia, so widespread. The problem is purification rates. And there are many similar alkaloids in extract, isomerase or conjugate. It seems to be a very time-consuming job to isolate. Probably due to that tough isolation work, the price is quite expensive. Irimaquinone belongs to melocespherinol. This one disrupts membrane, but there seems to be more biological effect. Isolation is easy. Latranquine A is a macrolide and it binds to actin and has been used for many years. The source sponge distributes widely from Okinawa, Indonesia, down to Vanuatu, Fiji, at least. You can find this sponge a lot on the underwater wall of Bunaken National Park at Manado, but sporadically at other places. In addition to these molecules, there are more compounds. So you can supply some bioactive molecules to researchers worldwide, and also it may be beneficial to some local researchers. But you need good isolation work together with instruments to confirm the identity and purity. By the way, probably most of the audience are not familiar with sponging. So if you do have an interest, please attend the invited lecture given by Dr. Nico de Groot early this afternoon, later this afternoon. The latter part of the second section is on the same or similar molecule from different source organisms. Manganese A is a unique alkaloid showing anti-malarial activity. Originally, it was discovered by my former colleague, late Professor Higa, and they coined the name after the collection site, Manza, in Okinawa. They reported this molecule from two sponges. The above sponge seemed to be quite endemic to Okinawa area. But the other sponge, Arkansas Strongylophora indians, is quite widespread. And I saw this Arkansas Strongylophora sponge in Indonesia at many dive sites as shown in these pictures. From Savan to Sorong, and also some down to South Kapa. Some of them do not look the same, but judging from the internal texture or morphology, I believe that they are the same. The chemical contents may be different between colonies. Actually, Professor Tsukamoto of Kumamoto University in Japan and her collaborators at INSULA reported some unique analogs as shown right. Another example is the two sponges. They look quite different, but containing the same molecule but different way. Particularly, copper sponge and microphysicians contain two more molecules named lorimalide and xamphanolide, both of which target microtubules. Therefore, biosynthesis genes are of interest by several research groups. However, it was not reported yet. As we can access both sponges in Okinawa, I supplied 3 specimens each to my collaborator to examine the genetic content. They found that there seemed to be not so much difference on the bacterial fauna for each species, but quite different between species. Also, we expect that the bacterial response for producing lactantrin can be found easily, maybe through we expected that horizontal gene transfer or same bacteria may live inside, but it was not that easy. Other one is remarkable. In addition to the previous sponge on the left, a different shaped sponge also contains the same molecule, but we have not worked on the right sponge at all. For halcomramide, there is a very similar looking Thai sponge containing the same class of macrolide, namely cabiramide. Also, cabiramide was found from Okinawa, originally from Okinawa, New Zealand. We have not identified which sponge contains the molecule. So, we have not examined the genome of cabiramide sponge yet. Cellular sponges. One of the most studied sponges in natural marine natural flora contain similar compound, namely misakinolide and sphenoide, with or without double bond at this position. Although the outer morphology is a little different, but internal texture are nearly the same. The genes for misakinolide was already sequenced years ago, but one for sphenoide was not reported yet. Although in the literature, cyanobacterial sphenoide was already reported. I heard that the same group worked for misakinolide, also worked on this sphenoide, but so far I have not seen their paper on sphenoide. Anyhow, on the origin of the unique metabolite, there has been a lot of discussion what kind of microbial sources or not. But there are only a handful of results on the biosynthesis genes of sponge metabolite. Summarize the second and the last part of my talk. We can supply molecules for life science studies from an orange. But there are a lot of unknowns how the molecules are produced in these orange. And the metabolite variation exists even within the same species. Okay. The last part of my talk is on the chemical and genetic diversity of two groups of organisms. First on the soft flora of the genus Phyton, and second on the Piliera nudibranch. Soft flora at shallow waters in coral reefs are easy targets for natural product studies from early stage of marine natural flora. And a lot of molecules, mainly diterpenoids and sesquiterpenoids, have been reported. Soft corals of the genus Phyton shaped like mushrooms, and they are rich in membranoid class metabolite, as shown here. About 30 years ago, I was asked to check one of the molecules named Phyto A as a source in Okinawa, because this molecule was reported to have anti-tumor promoting activity, and someone wanted to develop it also very critical thing matter we started. And we did a chemical screening on the software. As a result, the most dominant species was found to be consist of many chemicals shown here. Then we examined further on this group of organs with 3 viewpoints: chemical, morphological, and genetic diversity, together with biologists. As a result, one species, Sulphyton trochioform, collected out of Okinawa Island, is quite uniform in the 3 viewpoints. But the most abundant species, Sulphyton glaucum, was found to be quite complex, and it is likely to be consist of several clades or species as discussed by biologist Professor McFarlane in the United States. At that time, she already reported the species were divided into 6 clades. And more recently, Maloney in the United States and also LC, the collaborator of McFarlane, and our collaborators, including us, reported the chemical and molecular diversity of Parawan sulfite by analyzing mass spectral data of pure compounds and its lab with a platform named GNPS, Global Natural Products Social Molecular Networks, which is available to anyone. As a result, the species Sulphyton glaucum is consisted of mainly 2 clades: blue clade B and orange clade F, in addition to another species, Sulphyton trochioform. With GNPS works, the chemicals can be grouped into the right figure. And each circle represents molecule or related ions. And we may understand that some compounds can be found more in color blue or clade B, and the other in orange or clade F. It is not so clear, but it shows the complex nature of the species. Second group of organs we have interest are nudibranchs, cell-less mollusks, or sea slugs. When we worked on a few species as shown in this slide, we may understand that each species developed a unique chemical for their own details, and they developed it during evolution. And we looked into the most dominant species, most abundant species of the year in Okinawa. By collecting only 16 specimens, we examined their dominant chemicals and molecular phylogeny. As a result, one of the molecule, compound 1, was found from 5 specimens with the same sequence. So we expected that it might be the beginning of the differentiation of the species. To confirm this phenomena, we collected more specimens. After analyzing molecular phylogeny, the species were grouped into 5 species and 6 clades. And with chemical analysis, each clade may express a unique combination of molecules by showing the compound as numbers. But it is not easy to understand how they are complex with this figure. So my student also used the GNPS to analyze the diversity. As a result, the molecules are separated roughly into 3 groups. One is Becquerel type bicyclic compound. Second one is non-cyclic or other class of cyclic molecules, and one linear compound. And coloration of each circle indicates a clade. And you can see some compounds can be found only in a few clades. All right, but all for my talks, and I like to summarize. Indonesia has apparently has wide environment, and it's various places and explored places. So, and also as everybody knows, it is located at the center of biodiversity and for a triangle. So there must be a lot of bioresources in your water waiting for your research. And for marine nature product industry, it has passed at least more than 5 decades. But still gives a lot of chances for discrimination, not only new molecules, but new biological phenomena. And of course, everybody needs motivation to work on each field. It is, in my case, it is fun to observe unique marine organisms in the field and also discover new things in the lab. Okay. Thank you for your attention. That's all my talk.
Okay, thank you very much, Professor Tanaka, for interesting talk. And now we are moving to our next speaker, Tanaka Sensei. Hope you don't mind to wait before Q&A session. Thank you.
Yes.
Okay, our second presenter is Bapak Insinyur Mastur MSc PhD. Currently, Bapak Mastur is a president of Indonesian Biotechnology Consortium and head of research center for biotechnology and agricultural genetic resources. It's under Agency for Agricultural Research and Development, Ministry of Agriculture. He achieved his doctoral degree on bioregulation, Tokyo University in 1999, while his master and bachelor degree was from IPB University, Indonesia, in soil science and conservation, which is obtained in 1994 and 1988, respectively. Bapak Mastur will present in a video form, and please welcome Pak Mastur via video, who will be presenting the current research and biotechnology to enhance crop improvement in Indonesia. Okay, time is for Pak Mastur video.
Everybody. Today I will talk about the current research on biotechnology to enhance crop improvement in Indonesia. Agriculture sector is an important sector in national agricultural development in Indonesia. The sector is very strategic related to food security and agriculture-based economic development. Agricultural development requires support system from human resources, science and technology, genetic, land and water resources, agricultural input and infrastructure, and proper financial and policy system. In this term, agricultural development is directed to achieve advanced, self-sufficient, and modern agriculture. Agricultural development targeted to increase farm production and productivity, land area, and its supported system, production efficiency, which is supported by research, innovation, and mechanization. Indonesia has got target to achieve self-sufficient in several strategic commodities such as rice, maize, soybean, sugarcane, chili, salad, garlic, and cattle toward Indonesia feed the world by 2045. The ambitious target is important and required to set focus and synergy within resources to achieve the goal. However, there are several challenges in future agriculture, including expensive land and labor costs, water scarcity, and expensive costs, decrease on productive land for agricultural practices that force farming system on marginal and suboptimal land, and challenges on climate change impact on biotic and abiotic stress. On the other hand, high demand on healthy food lifestyle and environmental friendly also challenges people who work on agricultural biotechnology to develop crop varieties to meet this demand and challenges. Diversity of plant genetic resources, or PGR, is fundamental resource to develop a new plant variety or crop improvement. There are two basic conditions of agricultural practices that determine sustainable or not sustainable plant genetic resources in the future. At diverse PGR with normal income condition, development and commercialization of particular local superior variety without conservation activities in the region will lead to non-sustainable agricultural practices that will create biotic and abiotic problems in the future. On the other hand, involvement of in situ conservation by maintaining variation of local varieties in the area will bring sustainable production and commercialization of new varieties by crop improvement in the future. There are several in situ conservation methods that exist in forestry, for example, sagu plantation in forest area, or farming practices in arable area. The other method as a backup in plant conservation in ex situ collection, which applied in the field collection or in the seed bank. Indonesia has several institutes managing ex situ gene bank collection, both in the field and seed bank. ISIBIO grant collecting thousand accession of food crops, for example, rice, maize, soybean, sorghum, mung bean, peanuts, and other edible beans, root and tuber crop. In ISIBIO grant, we also have microbes collection and insect specimen. Other research institutes will gene bank collection. For example, some Indonesian research institute for rice, tuber, and beans, ornamental crop, vegetable, and fruit that cover about 17,000 accession. Plantation crops about 2,000 in total. As my previous statement, the biodiversity is important for crop improvement in the future. Fast development on biotechnology enables new techniques to develop new variety. Conference breeding requires long-time crop and enable molecular marker enhanced development of new varieties by molecular marker selection and new breeding techniques with application of genetic engineering by transgenic and genome editing technology. We fit incompatibility on intra or interspecies to create either what we call genetic modified organism or GMO and also non-GMO products. Other progress on in vitro study highlights inter-endosperm culture, protoplast fusion, and in vitro mutation on development of new varieties. Now I will start the topic on in vitro technology. There are three inventions that open pathway for development of in vitro technology. We can say that first invention to cell totipotency-based Swan and Sladen in 1838 was a pioneer for in vitro technology, followed by Harberland in 1902, who culture isolated cell, and invention in vitro germination media by Nathan in 1921. The current research in vitro technology, especially in ISIBIO grant, we develop micropropagation techniques for many crops, for instance, in root and tuber crops, horticultural crop, and industrial crop, which is some of them oil palm, sugarcane, bamboo, and some medical plants. Development of new improved superior variety has been released, for instance, on sorghum, rice, citrus, and chili. Application of in vitro technique, for example, in rice breeding, we utilize application of anti-culture to enable development of superior line from double haploid breeds, and combination of in vitro mutagenesis and selection to obtain new rice varieties for saline condition. Some new improved varieties using this method are Bioprima, Bioemas, Biosalin and 1 and Biosalin 2. Next, I will talk about the sequencing and marker assessment for breeding and crop improvement. PGR including modern cultivar, breeding line, local variety, land races, and crop relative species are very important as input into selection process through plant breeding to develop new and improved varieties. Out of species, small of high cultivated plant species were identified and food crops are at loss proportion. Therefore, it is important to set wise sustainable utilization of PGR to achieve food security using advanced technology, especially genomics. The genomic sequencing basically use DNA and also accompanied by RNA for gene expression that could specify a particular protein. All generated data from genome sequencing and further analysis will be for biological sequence of genome tests, and importantly, they are valuable and applicable further analysis. Why do we do genomic sequencing? With this technology, scientists are able to read genetic information. There are three types of genomic. First is comparative genomic. The second, functional genomic, involved in study on functioning and regulation of gene expression and metabolic pathway. And the third is structural genomic, which is determine complete sequence and gene map. Here are some examples of the application of genomics using several factors on crop improvement, such as genomic size, gene number, gene mapping and sequencing, evolution of crop plants, gene cloning, identification of DNA marker that can be developed as marker selection, transgenic breeding, linkage map, and ETL mapping. Importantly, this genome sequencing allows scientists for better prediction and phenotypic character on particular genotype called genome-based breeding. This improve and new varieties and related technology could be beneficial not only for human healthy, also for food safety, but also better environment. One important achievement is establishment of Pertanian Indonesia, as can be accessed in this link, icbigenome.litbang.pertanian.go.id, but in copyright in 2018. Genomic information of 11 species is featured the PGPI based on genomic sequencing and genetic mapping. Users are able to look genome browser of some crop with detailed information in genome. This PGPI provide variant for marker useful for DNA fingerprinting analysis and important source of molecular marker and gene governing desired traits. Marker assisted selection is main tool for molecular breeding. Especially genetic marker are linked to genetic variant affecting phenotypic change. Second process using molecular marker including single locus or genomic-based marker could be done in seeding phase, so it will reduce phenotypic selection and breeding cycle as well. Middle to high throughputing genotyping is notably for efficiency and reasonable cost and time. And GS is also a proof contribution for rapid development of functional marker as MAS to assist breeding. In case of marker-assisted background selection, this could be high-density molecular to detect QTL. ELJ also developed marker kit to detect early maturity of sugar palm that assist breeding and selection of local accession. In addition, several new varieties have been released and ongoing using marker-assisted selection estimated by Impari HDP, Impari Plus, Impari 40, Biopatenggang, and as essential variety. Marker-assisted backcross selection have been used for developing Bioni sestigera, which is resistant to brown plant hopper. Molecular breeding also surprised with total spiglet number character higher in biofortification of iron content on rice. And last technique, I will discuss distal genetic engineering, transgenic and genome editing. Transgenic, also known as recombinant DNA technology or gene manipulation, that alter gene in living organism to produce genetic modified organism with a new genotype. Various kind of modification are possible inside a foreign gene from one species into another species, genus or higher taxa, altering the existing gene to change the gene expression. Thus change genotype and phenotype, new product or variety. The genetic modification able to overcome sexual barrier incompatibility, and genetic engineering technology can be applied to improve product and environmental friendly. There are three steps genetic engineering research. First, gene cloning, where we can clone gene from unlimited gene source, including from different species, genera or kingdom. Decylated gen modified by molecular by addition of promoter and terminator to be constructed in expression factor. The second is gen transfer to introduce target gen 2. Direct transfer was applied with bombardment particle carbide silica, and indirect transfer was done by agrobacterium tumefaciens factor. The last step component is gen expression, where targeted gen cell be expressed and change the intended phenotypic character. There are several national institutions working on genetic engineering to produce GMO. For example, ISIBIO has been developed Biogranola agriorti, GMO potato resistant to Phytophthora infestan. Developed by collaboration ISIBIO and Cornell University. Also, drought research sugarcane already released by PTPN9. Other research are developed by insertion of alanine aminotransferase gene driven by root-specific promoter OSIRE. And the current status of this study in confined field trial to select baseline. There's also tomato GMO for GMP and TYLCV virus resistant, which is currently on multi-field trial CVT for food and environmental safety. The next technology is genome editing. This technology modifies gen or genome by inserting, replacing, deleting, and transferring precisely with the help to an endonuclease Cas9 enzyme as molecular scissor on specific target. Some example genome editing system are GFN, Talent, and CRISPR-Cas9. CRISPR-Cas9 currently is widely used and preferred because the system is more effective and editing application, drug discovery, and gene therapy to cure genetic disease. A pipeline for using CRISPR-Cas9 mediated genome editing to generate plant knockout mutant started by CRISPR-Cas9 construction and interrogated plant or known as plant transformation. Screening and regeneration of selected transformation were assessed, followed by genetic and phenotypic analysis on transformation plant. Genome editing system has been concerned in some university and research institute in Indonesia, not only ISIBIO, but also Gajah Mada University, Bandung Technology Institute, and also Indonesia Research Institute for Biotechnology and Bioengineering. ISIBIO already worked on the CRISPR-Cas9 genome editing system since 2016. Recent objective to obtain rice improved variety for semi-dwarf character and potassium efficiency. There are also CPVD virus-resistant citrus, increasing artemisinin content and chili resistant to Gemini virus. Other CRISPR-Cas9 genome editing research from Gajah to get albino or seed by mutation BDS3 achievement. Ministry of Agriculture, University such as ISIBIO, UGM, UNED, and research institutes such as LIPI also have been conducting research in biotechnology. Indonesia Institute of Science also obtained some new crop variety, either from soma clonal variation, marker resistant, irradiation, or genetic engineering technology. Biotechnology has contributed a lot on crops improvement and new varieties released to meet national program or human need and environmental friendly in general. ISIBIO has been released some improved varieties. From example, from marker selection, we released 5 rice and 1 chili improved variety. In vitro technology become great tool to produce 4 rice, 1 chili, 1 tangerine, and 3 sorghum new improved varieties. Regarding modern biotechnology research, Indonesia has released already drought-resistant sugarcane by UNED and Phytophthora infestan-resistant potato by ISIBIO. And from latest technology, CRISPR-Cas9 genome. That's all my presentation. Thank you very much.
Thank you, Pak Mastur, for a nice video talk. Okay, ladies and gentlemen, question for Bapak Mastur could be written in chat, and the committee will send your question directly to Dr. Mastur by an email. And I'm sorry about this. You can wait for the answer after the committee send an email to Dr. Mastur. Okay, so now we back to Professor Tanaka. Tanaka Sensei, are you there still?
Yes, I am.
Okay, to remind us on the lecture from Professor Tanaka, it was very, very interesting, Sensei.
Thank you.
There were three topics actually, and interesting one is the Professor Tanaka closing with remark that it is a big chance still for Indonesia to find new compound in the lab due to the unique location of Indonesia marine. Okay, so in chat we have 4 questions, Sensei. Yes. I will read. I will read. Yes, the first one is from Ibu Yakti, Pusat Studi Biotechnology UGM. I just wondering how you realize that you identified the new structure that have a potential to be developed in the future. Okay, maybe one by one, Sensei.
Okay.
Or
yeah, of course. I just replied short on the message, but I'm going to answer directly. Of course, when you want to develop the molecule into some other application or something, in that case, the original molecule is expected to have some biological activity usually. In case of, for example, halichondramide, it targets G-actin in our cells, and or just showing some toxic activity or antifungal activity. But in addition to that, it might be useful, for example, by attaching some kind of antibody. There is so-called drug group named antibody drug conjugate in ADC in short. So because of that, there is some easy to attaching point in the molecule structure. It might be useful if the molecule can be supplied in a synthetic biology method or recombinant biotechnology technique. But it's not it's a totally different field from my own. But it's not easy to supply. We better not think that we supply the molecule from nature, but by technology technique. Is that all right?
Yeah. Okay. I forward to number 2. It's from Ibu Tuti Murniasih. It's in Ancol. I think you visited there already.
Maybe Tuti is the one I met already many years ago.
Okay, yes. His question is, for compounds that have been applied as life science reagent, it is produced in synthesis process. Is there any most effective strategy for the selection of compounds from the isolation until applied?
Okay.
Sensei, we will have time until 11:28, and we still have 3 questions. So please yeah, be short and yeah.
Okay. Let me do. One example is that so-called domoic acid or kainic acid. Once a simple molecule like kainic acid, it has been supplied from nature. But nowadays, I believe some synthesis people have made a strategy. So it is supplied by synthetically in part. And also, but in the actual drug, like so-called eribulin, it is against breast cancer, and this drug is quite complex in structure. But since people also produce the molecule, because the in this case medicine is quite expensive, so you can it will be there is a reward for supplying by synthetically. So it is not simply on the structure, just by the structure, but the values of the molecule.
Okay.
Is that all right?
Can I read the third one?
Yep.
Yes. Okay. It's the question is from Ibu Tika. It's a researcher from BRIN. Thank you, Professor Tanaka, for the nice and interesting presentation. I have a question with regards to the diversity of marine secondary metabolites or bioactive compound. Are there any influence on specificity in the taxonomy, either species or family of respected organism, or do you think it was rather affected more by geographical or environmental factors or microbial symbiont? Thank you.
Okay. Judging from the molecular diversity, microbial resources, and also some among the cells-time organs, sponges are the most prolific sources of various molecules. In case of, for example, corals, soft corals, and also stony corals, they are not. They provide a huge number of terpenoids, but from the structural diversity, they are not really exciting. Okay, and what is it? Geographic and factors in microbiome. It's not that easy. We cannot answer easily. There are many cases, and in some, it was already known that some organs are quite different between colonies. And in other case, most any colony contains almost the same molecule. So, and there are, of course, some people examine microbiomes in bio, but it's not easy to conclude the fauna, microbial fauna in this kind of organism. As you can see in some of the literature, after obtaining so-called metagenomic DNA from sponges, you can find many groups of bacteria.
Okay, the last one is from Pak ISIBIO. It's about modification of the structure, the functional groups, or modifying the elongation of carbon bond. Are there any general rule or about steps that we have to consider first for modifying the compound structure? And the second one is a huge marine biodiversity exhibits a huge diversity of marine-based biochemical as well. So what is the current strategy in order to accelerate the discovery and development of drugs from marine organisms?
Okay,
So,
for the first question, in case of for some derivatives targeting protein, because of the huge size of the protein of the compared from the so-called ligand molecule, like natural product, probably we may need some we may need to attach some linker moiety, but I don't have much experience on this aspect, and it is better to consult other research papers when they target fish the target protein with the natural product. And for the second question.
Yeah, for the second question.
Yeah. Nowadays, big pharmaceutical companies do not prepare a natural product library by themselves, but small venture-like companies prepare. What I heard is we can provide library of natural products, and these companies screen against for some drug target. And find out the hit, I believe.
Okay, that's all, I think. If you have any question more, I think can we write an email to you, Prof. Tanaka?
What?
If the audience have any more questions, because we have a limited with the time. So can we send an email to you?
Oh, yeah, no problem.
Okay, thank you very much. Okay, thank you very much for Professor Tanaka and Dr. Mastur for addressing very interesting topics. I won't take a summary because it's very, very deep and well, I think it's very, very broad, also an interesting topic, and I think a lot of our researchers here is eager to actually make communication with you. Yeah. Okay, on behalf of the committee, I would like to thank to all speakers and to participants to the active participation. Thank you for joining us today, Sensei, and also for all of you, the participants. Now, I'm giving back the time to the MC. Yeah, I think the time is up and exactly, okay, thank you, Mbak Wibit. Thank you, Professor Tanaka. And
thank you, you are welcome.
Giving applause, big applause to the. Thank you.
Thank you.
Thank you, Professor Ecology, and also Professor Tanaka and Dr. Mastur for a very insightful session. Distinguished guests, ladies and gentlemen, we would like to remind you to please kindly fill out the attendance list of the first session that has been informed in the chat feature. Now, for the next agenda is Principle and Mechanism of Biotechnology Product session. We will hear presentation from Mr. Alfian Mauludin from Ditek Jaya. Mr. Alfian Mauludin, you would have 30 minutes and the time is yours.
Okay, thank you. Let me share my presentation first.
Sure.
Is it already up here?
Yes, it is up here already.
Okay.
The time is yours, Mr. Alfian.
Yeah, thank you. Okay. Assalamualaikum warahmatullahi wabarakatuh. Good afternoon, ladies and gentlemen. I'm very pleased to see you all. Thank you, and thank you for coming to this presentation. And really thank you to for give me this kindness of opportunity so I can be in here. Before I start my presentation and our discussion in this opportunity, let me explain to you and give the company profile for Ditech Jaya. Maybe I share first.
Excuse me, Mr. Alfian. The sound, we
Sorry?
cannot hear the sound. Maybe.
Oh yeah, sorry. Sorry. I
also
will
the sounds with the video.
share the sound. Okay. So.
Okay.
Okay, thank you for your thank you for your attention to see my company profile video. Before that, allow me to introduce myself. My name is Alfian Mauludin. I'm from sales engineer PT Ditech Jaya. As you know before, PT Ditech Jaya as a solo agent Shimadzu Indonesia. Okay, in this opportunity, I will share to you all about the analytical intelligence with Shimadzu solution. What is the analytical intelligence and what the Shimadzu solution for facing the next future maybe inquiry should be in the next future. Okay. Like we know, there are some difficulties in operation, such as diversification of working. Right? There are two types of diversification of working. The first is work efficiency, and then the second is variety user. Okay, as we know, day by day, the challenges in work are always increased, right? Such as the challenges for the increased job, many job or many tasks with a limited time. So what we need in here is how the instrument, how the technology can be support us to give them good efficiency, operator efficiency, right? And then for the variety user, there are two type skill operator. Is it like export operator? Okay, maybe no problem or no issue for the skilled operator or skill export operator, but how the beginner? For the beginner, maybe need the more technology or more help to that the task for this. How make any everyone, even beginner user, can use it the instrument as easy as they can. Okay. Then What the Shimatsu want to introduce to you all is it the analytical intelligence. Analytical intelligence in here means a smart analytical platform that incorporates state-of-the-art EOT. Is it like Internet of Things or everything about internet, everything about networking in here, and then analytical intelligence and machine learning algorithm functionality. So is it like machine we test machine to learning the algorithm function so that the machine can helpful to make our job, our task in laboratory especially to be easy. Okay, and then the analytical intelligence means to automation workflow include auto monitoring, auto controlling, auto diagnostic, and auto recovery function release the user from routine instrument operators. So when instrument have problem, instrument can be what I can say can be recovery without our help, something like that. And then what the purpose for analytical intelligence? Is it like for minimizing system downtime and then optimizing efficiency? When we get the minimizing system downtime and optimizing efficiency, so the productivity, our productivity will be increased. We will get the maximum productivity in our activity, especially in laboratory. Okay, next. What instrument have implemented this analytical intelligence? Okay, the first of thing is ultra high performance liquid chromatograph. Is it like UHPLC, right? And then as you see in my slide, in my presentation, is it the newest UHPLC from Shimatsu? We call it next era series. It means like next era or next generation era for facing any diversification of the working. Okay, this is type is it actually for modular system, so very flexibility for upgradable and as your own configuration and as your need in the laboratory. Okay, then. So what the intelligence in our HPLC, maybe you can see in here, this is like a routine workflow, which we always do in the laboratory, right? The first time we start up and then we do set up mobile phase. Is it like a important thing when we're using chromatography, and then we do column equilibrium, or we waiting until the system stability. So after system stabilize, we do analysis, start analysis. We inject a sample or standard in there. When the system running, we need to check, is it mobile phase check or is it enough or not? After that, we get a result and then we do system monitoring and a shutdown. And then we will using the instrument in next project or in another day. Okay, so how about the intelligent workflow? Actually, the intelligent workflow, like I showing in here, we can see every manual process can be run as automatically processed, such as auto startup. We can set instrument and accordingly what time we set the instrument will be turned on even we not yet arrive at the laboratory. Is it possible to do that? And then do mobile phase setup. So for mobile phase setup in here, we have a technology intelligence in here. We call it advanced real-time mobile phase monitoring. What is the advanced real-time mobile phase monitoring? Is it actually this feature for mobile phase setup? We have real-time mobile phase monitoring in here. We know the important things in HPLC. Some of them is mobile phase, right? When we not care about the capacity of mobile phase, it can be problem for our instrument and for our analysis. Actually, in here for mobile phase monitoring, is it like a sensor? Maybe I will show you in here. This is the sensor in here, gravity sensor. So when you put the mobile phase bottle in here, content mobile phase sure in here, so the system will be calculate automatically can be calculated automatically how much volume inside the bottle, right? Then we can set up the minimum volume for the mobile phase in here. So when the system decrease until the minimum volume which already we set, this it will be send the notification in case of shortage. So the mobile phase time monitoring can be installed and can be directly using in our smart gadget, our smart device, like maybe smartphone or something like that. And then what the purpose for advanced real-time mobile phase monitoring? Sure, is it for prevent system dry up? Is it like when we using the instrumentation and the mobile phase to be dry up? Is it will be impact to our column? So column will be easy to damage, and then another that the result or the analysis will be not good as well. So we need to reanalysis, so wasting time for this. So real mobile phase monitoring, as it means maximum of time. Okay, this is for one patent from Shimadzu for analytical intelligence. Is it advanced real-time mobile phase monitoring? Okay, then the next, after we make sure for mobile phase, then we do monitoring or column equilibrium in here until the column stable, until the system stable before we inject a sample or standard. For the conventional method, we do conditioning column with plasing instrument with set up flow rate, example like this. Maybe we set up maybe 1 milli per minute or more, something like that. When we first turn on the instrument, turn on the pump, will be make a pressure jump in here. If you using the higher flow rate, right, and then pressure jump in here will be impact to your lifetime column. Even that, so Shimadzu released analytical intelligence technology for flow pilot. Actually, flow pilot is it around the mobile phase flow rate gradually to set the point automatically. Is it mean when we set up 1 milli per minute in here, the system automatic run in a half of flow rate? Is it like 0.5 milli per minute in here, the example, and then when the temperature increase gradually, the flow rate will increase too. So this is prevent the extra pressure, so will be save your column lifetime. Okay. Another thing from the flow pilot protect column, is it everything automatically? You just set up in the software, and the software will be start your method, and then running the flow pilot protect column technology here. Okay. Then after we get the system stabilized, and then the column equilibrium, we got the good stabilization in there, we starting to start analysis, right? And then when we starting the analysis, we need to check the mobile phase, is it okay or not, enough or not, something like that, and then do system check. Maybe sometimes we get no good result when we do analysis. Is it can be because is it because maybe from the instrument, or maybe from our sample itself? Okay, so in the Shimadzu, they have another analytical intelligence. We call it auto diagnostic and auto recovery. It's mean like instrument can diagnostic if there are something problem in the instrument, but they can recovery too by themselves. Okay, so no need our help to recovery the instrument. Okay, what's the meaning in this slide is like this. When this normal pressure, right, when we do analysis in here, we inject, okay, in normal pressure, we will get the good result in here. But when sometimes the instrument or the pump, there are a bubble maybe in the mobile phase, so the pressure will jump the pressure will drop. So make a fluctuation in here. Pressure will be fluctuation. So when we inject in this condition, you will get no good result from your analysis, because that after we on this feature auto diagnostic, instrument will do auto purge to prevent this abnormal pressure. After auto purge, and the automatically system will give the normal pressure to exit normal pressure, and then conditioning again. After everything okay, system automatically will be re-inject your sample. So if the failure in the sample too, and then auto purge happen in here, the system will be re-inject sample to before. So this data will destroy and not saved in the database. So you will get the great result without you must waiting in the laboratory. So the benefit from this, yes, the chemical does not need to be physically present when abnormal condition appears. So after you preparation, after you start injection, you can go out, you can do another job, another task. Then you will get the result even in there. Next series can perform auto correction, ensure maximum system uptimes. Okay. Okay, and then after we do auto diagnostic and recovery, we got the result, and then we are reporting. So we calculate the result. We need to make sure if our instrument have a good maintenance schedule. So the Symposium solution give a smart service net. It means like some feature in our software, some feature in our chromatography software, like give the approximate maintenance timing by prediction from the use history. It means like when you must calibrate, when you must replace another part of like a consumable part or even spare part in there. You will not missing for the maintenance your instrument. If your instrument highly good maintenance, I think the lifetime is good to right, and then your result will be great. Okay. Is it that's all the simplest and intelligent workflow from Shimatsu? Is it our intelligent workflow automatically intelligent workflow in here? So important and so helpful for us in the next project. Okay. And the next slide, I want to introduce to you the newest gas chromatography. We already talking about liquid chromatography, right? Okay, so I want to share with you all about the gas chromatography. We call it next industrial standard GC generation. We introduce to you about a new experience, intuitive and simple controller like a touchscreen GUI, graphic user interface, and then what the meaning graphic user interface is mean like what the display in the GC instrument is it similar or same in the software too. So easy to make so easy to use this GC, and then we have new graphical icon improve operability. It's mean like the easy friendly use for your laboratory activity. And then we have innovative software enhancement like quick batch. Quick batch mean like when you are watching a movie in studio, the software will info you what the number via empty are already fill up, and then you cannot fill again if already taken by another file. Like similar like that. And then we have lab solution direct. Is it enable remote control and monitoring? Yeah, is it really useful when you're using when you working like a work from home? You can direct access. You can connect to your smart gadget, something like a smartphone or tablet. Is it another tools can be connected in there? And then the important thing in our new GC is tool-free routine maintenance. like I introduced you before, there are some talking about variety user rate. There are two type user. Is it a beginner and expert? This new analytical intelligence from Shimatsu purpose will help the beginner user to make everything easy. First, we need click tech nut. Is it like for replacing like a septum or glass insert? Maybe you can see in this tutorial video to make it easy to be understanding. There is just only need 30 seconds to replace septum or glass insert. We can compare with a previous technology. We need tools in there, so need time to replace it. So you can so the instrument another benefit is that save your time. And then how about the click tech connector in here? Click tech connector in here means when you install new column, so you don't need anything tools be used in here. Just only click tech connector, just open the GC and then you plug and play to the injector port. Something like this, so easy to beginner user. Like this, and then we have a smart jig. Smart jig, it's mean like a jig for make sure everything already according a GC requirement. Depend the detector you use it and the type of column you use it in the gas chromatography. So you need jig to measure how long the column installed to the injector port or to the column port, to the detector port. And then the simplest thing in new GC Shimatsu technology is it like a breeder oven. Breeder oven, maybe some people know notice for the lights we need inside the GC to get us when we do installation or when we do maintenance in the GC. Okay, Shimatsu already have a breeder oven inside the GC column oven. Okay. We have to clean gas flow line in here. The point is we just need a plug and play the filter when we do installation or when we replace a new filter in here. Okay, as you see in my presentation, you freely create your own configuration. Shimatsu give the flexibility for your configuration. You want to you want to create your own configuration, like such as you want to add with headspace system auto sampler, or you want to connect with thermal desorption or pyrolyzer or something like that. And actually, some of this configuration already installed in Brain. Or ISIBIO or another city. So in this slide, let me introduce to you all another product from Detect.jr products. We can offer to you. We can give your solution for your inquiry in the laboratory activity, maybe in the simplest like a spectrophotometer. We have a molecular spectrophotometer, UV-vis, even like from the double beam spectrophotometer until a single beam spectrophotometer. And then we have a Fourier transfer infrared, or we call it FTIR. You can see in here like a portable FTIR. You can move the FTIR to another place. Is it ER Spirit? And then we have a Huygens ER. Is it ER tracer can be upgraded until near. Okay, then we have spectrophotometer atomic. We have ACE. ACE is from like elemental analysis, tracer analysis. You can use it to measure elemental analysis from percent and up to the ppm. And then we have ICP and an ICP-MS. Is it more sensitivity for spectrophotometer atomic, which can measure until PPT levels. And then we another just we have a chromatography HPLC and GC, and then we have mass spectrophotometry such as like LCMS, single quad or triple quad, LCMS MS, and then we have GCMS single quad and GCMS triple quad too. And not forget, we have another solution for your unknown analysis. Is it QTOF MS from Shimatsu, which can combine with imaging imaging system IMScope. Is it in the process installation in LIPI? And then we have another product like thermal analyzer, like differential scanning colorimeter, and then TGA. We have another solution like for MALDI. Is it already have in LIPI2 in brain performance MALDI TOF in there. So we have a newest MALDI like a MALDI Mini and MALDI 8020 for the benchtop MALDI. We have another instrument like total organic compound for your environmental analysis. Is it like a simple instrument? Another simple instrument like balance, analytical balance, or top loading. Okay. The next is it like I want to introduce to you all. Beside the instrumentation, we have another solution for your laboratory environment. We call it a software solution. The challenges from this inquiry from every single laboratory is how to integrate the process with instrument become one solution. Is it we have a solution using a software. We have lab solution CS or client server like you see in here. We have lab solution CS which can connect more instrumentation in here. Even like not only chromatography system, we can connect a spectrophotometer system, and even we can connect another brand. So we under one platform software, we call it lab solution. So you can controller, you can reporting, you can do analysis in everywhere you want. As well, it in the same domain, in the same network. Okay. Like you see before, is it the newest digital transformation for your laboratory? Because another product, even like lab solution CS, we have a LIMS laboratory instrument management system. We have QMS, we have DMS, and Nickel training. So is it a full package for your digital transformation of your laboratory? Okay, this is the digital technology has been growing rapidly. So maybe we know we cannot notice if we need the technology as like analytical intelligence for our activity. Okay,
Excuse me, Mr. Alfian, you will have 3 minutes left. Thank you.
Okay, this is actually the last slide from
Okay, that's great.
Okay, so because we don't have much time in here, please, I want to make sure I want to point to you if you have another inquiry. Is it like not only you need an instrument, if you need the method or you need to maintenance or service or something like that, please don't hesitate to call me, to call my team, to call my company, to info our customer service in here. You can save my contact number in here, and you can reach my email in here in alfian@detectjaya.co.id or sales@detectjaya.co.id, and you can visit to our website. Is it detectjaya.co.id. Please don't hesitate to contact us in here. Okay, thank you, and see you, and nice to see you all in the great webinar today. Thank you. I will back to our moderator.
Thank you, Mr. Alfian Mauludin, for a great presentation. Ladies and gentlemen, before continuing to the next agenda, there would be some documentary videos from our partnership that we can watch. So let us watch the video together. Here we go.
Since 2019, PT Wadia Prima Mulia has continued to develop in meeting the needs of basic laboratories for Indonesian territories. Until now, our company has been trusted as an authorized distributor for 11 companies that produce the best laboratory equipment in the world. Our highly skilled and experienced sales team spread across Indonesia from Medan, Jakarta, Bandung, Semarang, and Surabaya. Our products. Sales services, including application and technical support, installation and training, also service and maintenance.
The next one.
In the life sciences, researchers are actively pursuing studies aimed at understanding life, the causes of disease, and achieving breakthroughs in regenerative medicine. To observe and accurately measure intracellular processes and structures, researchers need a microscope that combines sensitivity and speed. Increasingly, scientists require the capability to view cells and tissue at high resolution, as well as their larger context with macro to micro capability. All of these advanced features must be easy to use so that even novice users can acquire high-quality data quickly and simply. Olympus created the FV3000 series to satisfy these needs. This next generation of confocal laser scanning microscopes combines leading-edge imaging features with renowned Olympus optics.
All right, the next video. The next video for the host. While waiting for the host. Okay, here's the video.
Halo, kami dari PT ITS Science Indonesia. Hari ini kami akan memberikan penjelasan singkat mengenai pengujian porcine. Mengapa perlu melakukan pengujian porcine pada produk pharmaceutical? Beberapa macam bahan baku atau obat, termasuk tablet, kapsul, injeksi, dan sebagainya, dapat berasal dari produk hewani atau turunannya. Contohnya seperti gelatin, yang sering dipakai sebagai bahan utama kapsul atau stabilizer pada vaksin. Selain itu, antikoagulan seperti heparin juga dapat berasal dari porcine. Sedangkan berdasarkan Undang-Undang Nomor 33 Tahun 2014 tentang jaminan produk halal, produk yang masuk, beredar, dan diperdagangkan di wilayah Indonesia wajib bersertifikat halal. Oke, jadi bahan apa saja yang dapat mengandung porcine? Kebanyakan senyawa tersebut merupakan antikoagulan, digestive supplement seperti enzim lipase, amilase, insulin, dan sebagainya. Wah, cukup banyak ya. Tapi tenang saja, pengujian porsain ini tidak sulit kok. Ada beberapa metode yang dapat digunakan untuk mengecek kandungan porsain pada produk. Yang pertama, lateral flow device atau test pack. Metode ini cepat dan mudah tanpa perlu banyak peralatan laboratorium yang canggih, tetapi sensitivitasnya rendah. Metode 2, ELISA. Waktu preparasinya dibutuhkan 2-3 jam. Preparasinya cukup mudah dan dapat menguji dalam jumlah banyak dalam satu kali running. Dibandingkan lateral flow device, ELISA lebih spesifik tetapi masih memiliki batas deteksi sensitivitas yang sedang. Metode 3, real-time PCR. Metode ini adalah golden standard untuk pengujian porsain pada produk pharmaceutical dengan limit deteksi yang sangat rendah 1-5 kopi DNA. Pengujian porsain pada produk pharmaceutical sebaiknya dilakukan dengan metode PCR karena bahan baku yang diuji telah melalui proses pemurnian atau pemanasan tinggi sehingga kemungkinan yang tersisa hanya materi genetiknya saja. Selanjutnya, tahap real-time PCR ada 3 yaitu preparasi sampel, ekstraksi DNA, dan amplifikasi dengan menggunakan mesin real-time PCR. Untuk lebih jelasnya, mari melihat alat real-time PCR dari Roche, Light Cycler 96. Alat berkapasitas 96 well ini memiliki 4 channel fluorescence yang dapat digunakan sekaligus dalam satu kali pembacaan. Sebelum memulai proses PCR, profil temperatur dan protokol PCR dimasukkan ke dalam mesin. Setelah itu, tekan tombol start. Secara perlahan akan terbentuk grafik sinyal seperti ini. Sinyal ini terbentuk dari proses denaturasi, annealing, dan elongasi yang berulang, sehingga fluorofor yang terkandung di dalam master mix dapat berpendat dan tertangkap oleh sensor mesin. Sinyal yang didapat kemudian dianalisis menggunakan software Light Cycler, sehingga terlihat sampel mana yang positif atau negatif. Demikian penjelasan singkat mengenai pengujian porsain dari kami. Sekian dan terima kasih.
Now we will move on to the next video.
Halo, kali ini saya mau memperkenalkan alat laboratorium dari Syngene. Ingenious Tree. Sebelum pengenalan alat, apakah Anda pernah mengalami kesulitan saat mendokumentasikan hasil gel electrophoresis atau plot Anda di ruang gelap? Sistem dokumentasi gel secara manual terdiri dari ruang gelap, kamera, dan transluminator sederhana. Dokumentasi gel secara manual seperti ini kadang menyulitkan dan belum lagi kalau hasil foto gel yang didapat ternyata blur. Dengan Ingenious Tree, Anda dapat memilih pencahayaan sinar UV, sinar biru, atau putih, sesuai dengan pewarna yang Anda gunakan. Selain itu, software Genesis akan membantu Anda memilih pengaturan cahaya dan filter yang tepat, sehingga sangat memudahkan untuk mendokumentasikan gel fluorescence ethidium bromida atau cybersafe DNA atau protein gel dengan composite blue atau silver staining. Untuk penggunaan mode otomatis, Anda hanya perlu memilih tipe gel yang sesuai, kemudian software akan secara otomatis mengatur aperture, zoom, dan fokus serta posisi gel yang sesuai sehingga gambar gel terekam secara optimum. Software Genesis dilengkapi dengan database, tipe gel, dan pewarna yang tersedia di pasaran. Jadi jangan khawatir tidak bisa menemukan pengaturan yang sesuai. Sangat mudah. Hubungi tim ITS Science Indonesia untuk info lebih lanjut. Terima kasih. Stay safe.
And the last video.
Microtomy requires my full attention and dedication to make sure that I deliver the perfect slides. And with the Tissue Tech Auto section, I started getting the job done easier, faster, and with better quality. The automated block alignment makes sure precious tissue is preserved optimally. It is also a great help for making recuts, while the perfect alignment helps me produce even more slides. Trimming and sectioning are done with high precision, each section the same exact thickness. Thanks to the easy operation of the auto section, every technician in our laboratory is quickly up and running. Whatever the need, we are all confident to deliver the perfect slide each time. My name is Mark Shanti, and I'm empowered by Sakura.
All right, ladies and gentlemen, for the next, we will hear a presentation from the Director of CropLife Asia, Mrs. Soni Tababa. Mrs. Soni Tababa, the next 30 minutes is yours. You may proceed.
Thank you very much, Ibu Wiwit. Let me first share my screen and see if, are you able to see my screen now?
Yes, we are able to see it clearly.
Okay, let me just put that into slide mode. And can you hear me well?
Yes.
All right, thank you very much. So I'm standing with, not really standing, I'm sitting right now, but I'm just the last speaker and your break is coming soon. But first of all, I'd like to thank the organizers of ISIBIO 8. This is a lot of work. Congratulations to BRIN and to KBI putting together all this conference, all the researchers with all this fantastic work they have done. And annually at that, since 2012, is really a lot of work. This is, CropLife Asia has a similar, I manage a similar program for 15 years annually, and it's a lot of work. Very much big congratulations. And the other one I'd like to say is that I just would like to thank you for our organizers and participants for allowing us, giving us a space to share our thoughts around plant science, around innovations, as well as around food security. And in the next few slides, before I go to the next few slides, so I am Ibu Soni, and right now I am in Philippines, in Los Baños, Laguna, but I work as a Biotechnology Affairs Director in CropLife Asia, which is based in Singapore. So the pandemic brought me another blessing to be with my family, spending more time with my family while working from home. So let me go next to my next topic, which is just to give you a flavor about CropLife Asia and its network. I always would like to introduce first CropLife Asia. We are a regional organization, and our global organization is CropLife International. CropLife Asia is based in Singapore. We have 15 national associations all the way from North Asia to Southeast Asia and down under. And you can see on your screen the 6 global R&D companies that are our members, and they are very much active in crop protection, seeds, and plant biotechnology space. And I said we have 50 national associations. You see them on screen, and I'm very happy that in Indonesia we have CropLife Indonesia, which have been in Indonesia helping and contributing to societal goals in Indonesia is CropLife Indonesia, more than 3 decades there. And for our biotech program, you would notice that some of our national association focus mostly on crop protection. So for biotech and seeds, we partner with other organizations like in India, the Federation of Seed Industry of India, where they have a special interest group, which is Alliance for Agri-Innovation in India. We have the Council of Biotechnology Information in Japan, a CropLife international affiliate, and also China is so big that we have to kind of separate the program from a crop protection, purely crop protection, and for biotech. And as you see, we have 15 national associations here. Most of CropLife bring together the advocacies on not only crop protection, but plant biotech and seeds, but others just focus on crop protection. And CropLife associations, whether they are in the region or whether it is CropLife Indonesia, our vision is really to enable a productive food and agricultural systems through innovative crop science and technology. The vision is to have a living standards and improve living standards for not only our farmers, but for those who are using, who are involved in farming in a very sustainable way, and ultimately to improve and contribute to the important goal of food security. And so you would notice that our company members are really into developing innovations in crop science. So the major stakeholders where these technologies will be accessed are by our farmers. So we are hoping that our farmers will be able to grow more on less area through these innovative technologies, so that later on you could see that growing more in less area would help in our advocacies and our goals, overall societal goals in biodiversity conservation, and even in reducing the impact of climate change or in mitigating climate change. So just as a background here, CropLife, whether we talk of CropLife Indonesia, we have programs around crop protection, plant biotechnology, seeds, public affairs. Stewardship is very important. Stewardship is responsible care and anti-counterfeiting. And, you know, when it's a good product, it is likely to be copied. So we have to be very careful as well and make advocacy around anti-counterfeiting so that our farmers would have the guarantee that the products that they are using are genuine and would be able to benefit from it. So I'll just focus my next talk on global events in 2021. I think what I can say about 2021, there are so many big events that impact the way policy will be crafted in the next few, maybe in the short term, 3 years. But this will also impact us being scientists, or even from the industry. So let me just talk a little bit about COP15. So COP is a big police, but it stands for Conference of Parties. And this is about the Convention on Biological Diversity. So it is supposed to have been held in 2020 because this is held every 2 years. But because of the pandemic, this was started to be held in October this year. A high-level segment was started in China, in Kunming, and the theme is ecological civilization. So as you know, this is Convention on Biological Diversity, but this Convention on Biological Diversity is about conservation, about the use of biological diversity, and also benefit sharing. And so we have subsidiary agreements. So you must have heard about the Nagoya Protocol on the Cartagena Protocol first on biosafety, which is the only legal agreement around living modified organisms. You have the Nagoya Protocol on access and benefit sharing, and you have the Nagoya Kuala Lumpur Supplementary Protocol on liability and redress. So as you would note here in Kunming this October, last month, the high level, the parties to this called for urgent and integrated action to reflect biodiversity conservation in all sectors of the global economy. So whether you are in trade, so everybody is enjoined to really be accountable for biodiversity. So it's not limited to a few actors. And there's a call for increased investment and more private sector involvement. So there's a website here. This is where you can get updated news around what happened to COP15. This is a continuing, there's an ongoing negotiation that will be held in person next year in January in Montreal. So probably there will be some coming from Indonesia as party to the Cartagena Protocol to negotiate in this conference, in this convention. The other one I'd like to call is the UN Food System Summit. This one really focused on our food system so that we are called to deliver not only on the 17 Sustainable Development Goals, but really asking member states, the parties to this, to account, to make a reporting. And so, and make more solid commitments towards more sustainable, equitable food systems. So before the summit that was held in September, there were many independent summits and also summit by member states. In fact, as early as January, Indonesia held that summit around the Food System Summit. And so that is very important because maybe in the next 3 months or in beginning 2022, we will hear our leadership in different ministries to talk and set directions around our food systems, around our biodiversity. And the last one I talk is about, I will mention is around the Climate Change Conference, which recently happened in UK, in Glasgow. So this one really, they put together solid commitments. All the parties, they were asked to make their commitments instead of 2040 2040, move it earlier so that you can have global net zero emission. And the other point of this climate change conference is they also mention agriculture as making it more resilient to climate change. And if I just want to bring to your attention, just in case it has missed into your radar, that last month, the ASEAN adopted the framework for circular economy. And so I put together the strategic priorities. It's around harmonization and mutual recognition. And we are talking now of circular products and services, around trade, around innovation, finance, so energy. And so these are, it encompasses a large sphere of the things that we are doing, and it will impact also the way research will be done and the directions of research that will be done, whether you are in the public sector or in the private sector. And we heard Vietnam is already, the Minister of Agriculture in Vietnam is already very interested in discussion about circular agriculture, and Thailand is also very interested. I have yet to hear from Indonesia how is the discussion around circular agriculture in particular. But it's very interesting. This is something that, everybody must be engaged. And so if we talk around, we go back, we remember Convention on Biological Diversity, Food System Summit, the climate change, this circular economy. I'll just highlight this common thread, not just the three of them, but for this talk, I'll just highlight the commonalities among those developments around the important role of agriculture, of innovations, technologies, and of partnerships. And this is so later on, I'll talk about that, but let me just segue a little bit and go back to the farmers, because whether it's climate change or whether we look at the food, every time we eat, we should thank the farmers. And because they are the frontliners, they are our heroes, they produce our food, but they are facing a lot of challenges, whether it be from climate, because climate could bring extreme weather, more rains or more severe drought, or more pests or changes in weeds and more diseases. And so these are a lot of challenges that farmers have to face, and we are also asking him, hey, produce more, produce sustainably. And so there's a lot of responsibilities that we bring on to the farmers, but we should also be in a position to be able to help the farmers, and that's why we're having this conference. So a lot of the disciplines that will be presented here will be able to find their way to farmers' hands. I'll just go back. This may be a 2014 research from the International Food Policy Research Institute, but this just highlights how important the contribution of technology in Indonesia. So whether they be in crop protection, they can increase yield as high as 20%, or in having crops that make use more efficiently of nitrogen, or having drought tolerant or salt tolerant crops. So when there is extreme weather as a result of this climate change, we see more the technology as a very important intervention to help mitigate or adapt in climate change. So this is another example, no-till. So for example, no-till help in not only in carbon sequestration, but it also double the maize. So if you don't till, you help in carbon sequestration, but it also help in improving yield. And this is the data again coming from the IFRI, which is a think tank research agency under the consultative group of international agricultural research. Now, just zeroing on some of the technologies in supplying food sustainably sustainably, particularly from a plant breeder's perspective, they can do a lot. They can develop varieties that can protect or reduce insect damage. They can develop novel herbicide tolerances or disease-resistant varieties, as mentioned with Master earlier, improvement of varieties that are more resistant to pests and diseases. And then we can also develop varieties that can, or farming systems that can improve carbon sequestration or improve tolerance to salinity, drought, or even improve the nutritional profile. And with the advances in science, we see, and we have been hearing, you are all scientists and researchers, so you have heard how novel molecular techniques and transgenics help in that direction. So Master earlier said that I'm very happy to hear that GM potato, earlier there was GM sugarcane, and now GM potato is now advancing, and maybe very soon we can see it really in the market after the variety registration approval of GM potato, and that is very good. And now we also heard from Master the gene editing, and so those are very important innovations. Using these techniques to improve the varieties is a very important way of helping farmers cope with their challenges. I am in my last slide, but this is where I would like to, I would say this is a distillation of my so many years already. I started working for government in 18 years with the Department of Science and Technology in the Philippines in R&D management, and then move on to a regional agency for agriculture under the ISIBIO, in communicating plant biotechnology. And this slide seemed for me like a distillation of, also I share the same thought of what are the elements in the environment that could enable plant science innovation. Not only enable, but you would have timely access. I think timely and access is very important to that. So you would see here a lot of bullets, and I just would like to say that these are not exclusive. They impact on one another: political will, science and evidence-based regulation, societal license. So for example, when I worked in the Department of Science and Technology in the Philippines before, then when we tried to craft the crop biotechnology research focusing on our Philippine export winners, and then rice and corn, we have to reach out to our policymakers, not only to our heads of agencies, but even the legislative. We have to make sure that regulations are there, and also risk-based and science-based. And just one thought about regulations, from a biosafety point of view, we have regulations. So for example, Indonesia, they have the regulation as early as 2005. And so over those years, Indonesia has gained a lot of regulatory experience. And with that regulatory experience, you can get to a more comfortable and confident level how to make it more efficient. Societal license is very important. If consumers, so like the GMOs, there's until now, you can still hear a lot of people against it. You can still hear a lot of misinformation. So, and that is making our politicians nervous as well. And that is also making our regulators, slow down or just kind of take a little bit time, because, when there's a lot of noise, that is also affecting the decision making. But here we really have to also listen to our farmers, because farmers need choices. And you are researchers, and not only researchers, when you do into commercializing the products of research, we have to increase our investment. Public investment to research must be sustained and must be increased. IP protection, intellectual property. This is very important because when you invest in research, you also want to gain something from your investment. And the last one here is public-private partnership. Whether it's a global event or global agreements, public-private partnership is important because nobody can exist as an island. And so we're hoping that CropLife Indonesia has been, all these elements, they've been working on this, engaging our governments, engaging the different stakeholders, that someday we can add more choices to the crops that are available for farmers, not just GM sugarcane, not just GM potato, but also GM corn and the other, I heard from Pat Master, the GM tomato and all the other products in research still on gene editing. We really hope that those can come on the ground commercially, that it can be commercialized, be at the hands of our farmers, and inevitably that will redound to the benefits of consumers. So that will be my last parting thought. And with all your help, we can all live in a better planet. Thank you so much for giving us this opportunity.
Thank you, Mrs. Sonita Baba, for the great presentation. Ladies and gentlemen, now we will have a break for an hour and we will start the parallel session at 1:30 p.m. Jakarta time. Thank you.
National Research and Innovation Agency, a synergy in an orchestra. Established through the Presidential Regulation Number 78 of 2021 about the National Research and Innovation Agency. To enhance science and technology's contribution to the national development, and to improve the research and innovation ecosystem in Indonesia. The National Research and Innovation Agency is a government agency that is directly responsible to the President of the Republic of Indonesia. Consists of 7 deputies, supported by various research institutes, and have more than 70 research centers and supporting units. Through the 3 directions: science and technology resources mobilization, providing a favorable research ecosystem, building an economy foundations based on research. The presence of National Research and Innovation Agency is to achieve Indonesia's goals as a developed country in 2045. Also to make Indonesia as a global reference for sciences, bringing advantages on science and technology development, policy making, and societies.
Halo sahabat LIPI, selamat datang di video tour Biosafety Level 2 dan Biosafety Level 3 LIPI. Lembaga Ilmu Pengetahuan Indonesia atau LIPI merupakan lembaga penelitian yang memiliki fasilitas laboratorium Biosafety Level 2 atau BSL2 dan Biosafety Level 3 atau BSL3 yang terletak di kawasan Cibinong Science Center, Bogor. Laboratorium ini digunakan untuk melaksanakan kegiatan penelitian yang melibatkan organisme dan patogen yang berbahaya. Saat ini, kedua laboratorium tersebut telah ditetapkan sebagai laboratorium pemeriksa COVID-19. Biosafety Level, atau biasa disingkat BSL, adalah suatu tingkat sistem keamanan yang dibuat untuk menangani bahan biologi berbahaya di laboratorium. Tujuannya untuk melindungi personil yang bekerja di dalam laboratorium juga lingkungan dan komunitas sekitar dari paparan mikroba patogen yang sedang diteliti atau diuji. BSL memiliki 4 tingkatan, dari 1 sampai 4, di mana semakin tinggi tingkatannya, maka semakin khusus fasilitasnya, dan semakin berbahaya bahan biologi yang dapat ditangani di fasilitas tersebut. Perbedaan utama dari tingkatan ini adalah sistem tata udara, tekanan, kelengkapan fasilitas, dan alat penunjang lainnya. Kita ke laboratorium BSL-2 dulu yuk! Di dalam BSL-2 ini, ada banyak ruangan yang digunakan sesuai fungsinya untuk deteksi virus SARS-CoV-2 sebagai penyebab pandemi COVID-19. Ruangannya terdiri dari 1. Ruang penerimaan spesimen. 2. Ruang logistik sebagai tempat penyimpanan bahan kimia dan bahan habis pakai. 3. Ruang ganti untuk pemakaian alat pelindung diri. 4. Lab ekstraksi yang terdiri atas ruang antara, ruang laboratorium, ruang mandi, dan ruang dekontaminasi limbah. 5. Lab PCR yang terdiri atas ruang antara, ruang pre-PCR, ruang post-PCR, dan ruang analisis. Nah, kalau tadi sahabat LIPI sudah ke BSL-2, yuk sekarang kita ke BSL-3. Fasilitas laboratorium BSL-3 LIPI memiliki infrastruktur yang lengkap dan telah tersertifikasi sesuai standar WHO sejak tahun 2018 dan terus diperbaharui setiap tahun. Biosafety Level 3 memiliki 4 laboratorium, yaitu Clean Rooms, yang terdiri dari Health Biotechnology Laboratory, Food Biotechnology Laboratory, Biosafety Lab, terdiri dari BSL-3 Laboratory, Animal BSL-3 Laboratory. BSL-3 LIPI terdiri atas 2 ruangan. 1. Ruangan mikroba. Ruangan ini digunakan memperbanyak atau mengisolasi materi genetik bakteri untuk digunakan dalam kegiatan penelitian, misalnya pencarian obat baru, vaksin, dan sebagainya. 2. Ruangan kultur sel. Ruangan ini digunakan untuk mengkultivasi virus di dalam sel mamalia hidup. Sedangkan Animal BSL-3 terdiri atas 2 ruangan. 1. Ruangan ISO cages. Ruangan ini digunakan untuk memelihara hewan pengerat yang digunakan sebagai model pada uji di laboratorium. 2. Ruangan pembedahan. Ruangan ini digunakan untuk melakukan pembedahan hewan dan mengambil sampel organ tubuh dari hewan. Nah, untuk mendukung pemerintah dalam mempercepat penanganan COVID-19 di masa pandemi, ruangan di Animal BSL-3 ini dialihfungsikan untuk mendeteksi COVID-19. Sehingga, semua ruangan di lab BSL-3 digunakan secara aktif untuk semua kegiatan deteksi dan riset SARS-CoV-2. Seluruh ruangan di BSL-2 dan BSL-3 dilengkapi dengan berbagai peralatan yang mendukung aktivitas penelitian, juga didukung oleh sumber daya manusia yang terdiri atas para peneliti dan prekayasa terlatih, tim logistik, dan tim administrasi. Melalui fasilitas laboratorium BSL-2 dan BSL-3 yang dimiliki, LIPI siap berkontribusi secara maksimal untuk mendukung pemerintah dalam mempercepat penanganan COVID-19. Sahabat LIPI dimanapun berada, jangan lengah, jangan menyerah, dan jangan lupa untuk selalu mematuhi protokol kesehatan dimanapun berada ya. Bersama, kita bisa melawan pandemi ini. Salam sehat dan semangat ya!
divided into 3 breakout rooms. Room 1 and Room 2 will begin with the invited speakers. Room 1 with Professor Anuraga Jayanegara with the topic Modification of Polyunsaturated Fatty Acid Metabolism in the rumen and their deposition in animal products by dietary polyphenol. At Room 2, we'll have Dr. Yopi with the topic Center of Excellence for Integrated Biorefinery as a platform for developing Indonesia's biotechnology research in the future. And at Room 3, the session with the invited speaker will start at 2:30 p.m. Jakarta time with Professor Nicole J. De Vogt with the topic Sponge as Essential Elements in Tropical Reef Ecosystem. Parallel Session 2 will start at 2:50 p.m. and the Parallel Session 3 at 5 p.m. Jakarta time. And all the presentation of parallel session are scheduled according to the latest rundown shared by the committee. And to enter the room, choose the square icon, breakout room, on the toolbar below and click join next to the room that you choose. Now you might enter the breakout rooms. Thank you.
Mbak Wiwi, saya di nomor berapa ya? Mbak Wiwi? Halo?
Center in parallel session 1. First, I would like to introduce our invited speaker, Professor Dr. Anuraga Jayanegara. He is lecturer and researcher at Department of Animal Nutrition and Feed Technology, Institute Pertanian Bogor University. He is also as guest lecturer in Hiroshima University, My University, Jen University, Poznan University of Life Science, and also an ETH Zurich. He got his PhD in Animal Nutrition, Swiss Federal Institute of Technology, Zurich, Switzerland at 211, Master in Agriculture Science in Tropic and Subtropic University of Hohenheim, Stuttgart, German at 208, and Bachelor in Animal Nutrition and Feed Science at Institute Pertanian Bogor University at 203. He has produced many publications about feed science, animal nutrition, ruminant, poultry nutrition, and he has been recorded to an high index as 23 and Google Scholar. And the awesome one, he was the first national outstanding lecturer in the science and technology category by the Minister of Research, Technology and Higher Education at 219. Without further ado, I would like to welcome Professor Dr. Anuraga Jayanegara. The time is yours.
Thank you very much, Mrs. Chairwoman, sorry, to Dr. Evi Susanti for the time given to me. Please allow me to share my screen. Is it visible now?
Already then.
Okay. Okay, hopefully everything is fine. Can you see my slides?
Yes, already.
Okay. Thank you very much, Ibu Evi, as the Mrs. Chairwoman, as the moderator for today. Okay, good afternoon, everybody. Assalamualaikum warahmatullahi wabarakatuh. Please allow me to deliver my presentation, my topic on the modification of PUFA. What is PUFA? PUFA is polyunsaturated fatty acids, metabolism in the rumen, and their deposition in animal products by dietary polyphenol. So I'm from IPB University. So I have 6 outlines for this presentation. The first one is introduction, and then a little bit mechanism on lipolysis and PUFA biohydrogenation, and then how to, I'm going to show you some strategies for manipulating PUFA biohydrogenation in the rumen, and then start from polyphenol and its influence on PUFA biohydrogenation, and then PUFA polyphenol and PUFA deposition in animal products, and finally a short description on the multi-functionality of polyphenol with related to animal nutrition. So nowadays, if we talk about animal production system, previously if we consider animal production, it was merely concentrated on how to produce a huge or high productivity of animals, more live weight gain or more milk production per unit of animal. That was in the past, but also nowadays. Yeah, so focus on animal production. But now it has been expanded. Nowadays, if we talk about animal production, we need to consider also so-called the environment. So animals, particularly ruminants, they emit methane, one of the biggest source of greenhouse gas emission after carbon dioxide. This is also an important aspect to be considered in animal production system, especially from ruminants. And then the last one, which is I'm going to deliver for today, is how to improve the quality of animal products, how to deliver a nice, healthy animal products like meat and milk. So this is my focus for today's presentation. So with related to animal products, meat and milk, one of the important aspects is the fatty acid profiles, or polyunsaturated fatty acids. So they are considered to be beneficial for human health. We want, of course, in order to deliver positive health effects for human, we want to increase the PUFA, especially the omega-3 fatty acids. One of them is alpha-linolenic acid. This is here we want to increase this. And then also the so-called conjugated linoleic acid, or CLA. The main isomer of CLA is ruminic acid, cis-9-trans-11-C18-2. And this is considered also regarded as fatty acid that possess anti-cancer activity. So these 2 components of PUFA we want to increase. But then there is also fatty acids that are regarded to deliver or to give negative health effects for humans, especially the saturated fatty acids. Yeah, if we talk about C18, then it is C18-0 or stearic acid. And of course, we also know that cholesterol, this one also negatively affect human health. So basically, we want to increase this. And at the same time, or simultaneously, we want to decrease this. So how about the lipolysis and PUFA biohydrogenation? So this will give us a strong background in order to understand what will be the research found. What is eaten by the animals actually the feeds, and feeds contain a number of nutrients. If we talk about macronutrients, they are consisted of carbohydrates and protein, and also the lipids. Yeah, lipids, when they enter the rumen, it will undergo so-called lipolysis. Yeah, I'm going to show you after this lipolysis, and then after lipolysis, then the second step is biohydrogenation. So basically, lipolysis is a prerequisite or a requirement in order biohydrogenation to occur, and then finally it will produce the saturated fatty acid. So basically, these are the saturation of unsaturated fatty acids, the removal of double bonds, yeah, by hydrogen in the rumen. So what is lipolysis? Lipolysis actually is the liberation of the fatty acid. Yeah, previously it is ester linked with the glycerol backbone, and then after lipolysis, it is liberated to produce the free fatty acid. Yeah, and then this free fatty acid, if it is unsaturated, then it will undergo the biohydrogenation. The biohydrogenation itself is really a complex process, and there are a number of pathways linked to this biohydrogenation. If we talk, for example, the linoleic acid, C9, cis9, cis12, C18-2. So it has two double bonds. Initially, it will be transformed. The cis12 will be transformed to trans-11, and then this trans-11 is so-called the ruminic acid, the CLA, conjugated linoleic acid. And then it will be converted to phexanic acid with only one double bond. This has two double bonds, this one, and finally will produce stearic acid, which is the saturated fatty acid. Similarly, this is the linolenic acid. It has more steps, yeah, until reaching the final or the terminal product of the biohydrogenation, because it has three double bonds. And who is the actors? Yeah, who are the actors responsible for this biohydrogenation? Actually, there are some classes of microbes responsible for this. Yeah, but the main or the most well-known microbes, there are two. Yeah? The first one is the Butyrifibrio fibrosulfans. This one, yeah? And the second one is Butyrifibrio proteoclasticus. This one, this fibrosulfans is responsible for the initial step of biohydrogenation, while the second one, this is responsible for the terminal or the last step of biohydrogenation, converting the phexanic acid into the stearic acid. So what about the fatty acid metabolism in animal products or in the animal tissue, like in the milk or milk? Yeah. What is happening in the rumen is actually the saturation. Yeah, the biohydrogenation, or in another word, is the saturation of unsaturated fatty acid. But in the mammary gland and also in the meat, yeah, there is a vice versa, or the reverse order of this biohydrogenation, so-called desaturation. So this, for example, phexanic acid from trans-11, it will be converted back to ruminic acid. And this happen in the mammary gland or in animal products, yeah, and also in the meat of ruminants. So if we try to deliver the phexanic acid into animal products, into the mammary gland, into the tissue of the animals, into the meat, yeah, then it will be to a certain degree will be converted to ruminic acid, which is beneficial and it has anti-cancer property for human. What about the strategies for manipulating the PUFA biohydrogenation? I've listed a number of principal strategies. The first one, of course, we need to increase the PUFA supply. We may choose oils or lipids which are high in PUFA. For example, the fish oil yeah, or algae, oil from algae. So there are numerous options of increasing this PUFA supply right directly from the feed sources. And then afterwards, we inhibit the lipolysis, yeah, because this is a prerequisite for the biohydrogenation to occur. Yeah. And we may specifically targeting this microbe, the anaerobic Vibrio lipolytica, the main responsible microbes for lipolysis. And then inhibiting the butyrophibrio fibrisulfans, increasing CLA concentration, increasing the taxanic acid, because it will be converted back to delta-9 desaturation activity in the mammary gland or in the animal tissue. And finally, we may inhibit the terminal step of bioregeneration. It is the butyrophibrio proteoclasticum. So these are the principal strategies, different steps that we may inhibit the biohydrogenation. And we come now start with the polyphenol and its relationship with the PUFA biohydrogenation. Why we are interested in polyphenol? Actually, polyphenol itself is a natural compound. Yeah? There are different classes of polyphenols in different kinds of plants. Yeah? So actually, the chemical structures of polyphenol are very diverse. And it has no residues because, of course, this one is a natural. Yeah, it is not a synthetic compound, different with some other compounds. And more importantly, it is abundantly available in the tropics. So our plants have a number or a high concentration of polyphenols. I had an experience when I did my PhD in Zurich in Switzerland. So there I compared between many tropical plants and temperate plants forage. Yeah? And so the tropical plants contain polyphenol double or triple or even more as compared to the temperate plants. So we are rich in polyphenol, yeah, our condition, because polyphenol itself is synthesized at or during the stress condition. For example, at high light intensity, high infestation from predators, yeah, both macrofauna or microfauna or microbes. Yeah. So naturally, polyphenol is synthesized as a defense mechanism against the unfavorable condition occurring to the plants. It has antimicrobial property and it has also multifunction. Yeah. So this is the reason. These are the reason why we choose polyphenol. So phenolic can be divided into two main fractions. The first one is tannin phenolics, and the second one is non-tannin phenolics. The non-tannin phenolics is a more simple phenolic. Yeah, it doesn't bind to protein, while the tannin phenolics, it binds protein. So it binds protein, and within this tannin phenolics, it can be divided into two main groups. The first one, the hydrolyzable tannin, and this is easily or more easily degraded by rumen microbes, yeah, under anaerobic condition to generate this simple phenolics and also the sugar. While the condensed tannin, it's hard to be degraded under rumen condition, under anaerobic situation. So it can be degraded, it can be it can be broken, but under aerobic condition, it needs more energy to be degraded. Okay, so this is the typical chemical structure of hydrolyzable tannin. It has a core of glucose, while in this condensed tannin there's no glucose or no sugar at all. We have examined a number of tropical forages and their tannin or phenolic concentration. Yeah, as I mentioned before, that the concentration are much higher as compared to temperate. Yeah, so the tropical forages, yeah, contain much more phenolics or polyphenols as compared to subtropical and temperate forages or plants. And in general, also legumes contain more polyphenol as compared to grasses. Okay, now we come across to the fact or some research results showing that polyphenol is able to modulate rumen biohydrogenation of unsaturated fatty acid. So the first one is the ability of polyphenol, to decrease the lipase activity. We see here that when the phenolics, the polyphenol is higher, then the lipase activity is lower, because polyphenol is able to interact with protein and including the enzyme. Lipase is an enzyme, it is a protein, mainly the protein, although it has cofactor or coenzymes, but mainly they are protein. So it interact with the polyphenol and become less active as compared to the intact form. So here, so
Excuse me, Prof.
yes,
We have less
already.
than 2 minutes.
Okay, I will speed up my presentation.
Thank you.
And if we look at the correlation between polyphenol and the unsaturated fatty acid, we could see here. So increasing the polyphenol then reduce the disappearance of omega-3 fatty acid. Here is the alpha-linolenic acid as well as the linoleic acid. It is the same message, and this is also the same. Increasing phenolic content reduce the biohydrogenation of C18-3. And here we can see also that phenolic reduce the activity of Bacteria fibrosolvents, the main microbes, one of the main microbes responsible for biohydrogenation. This is also the proteoclasticus, again, reducing also. Okay, it increased the CLA, the total phenol. Tannin also increased the acetic acid, as I mentioned before. This is also showing us the ability of tannin to reduce the trans 11, the vaccinic acid, increasing, and simultaneously reduce the stearic acid here. Decreasing also the saturated fatty acid. So we can see here that tannin is, or polyphenol is consistently reduce the lipase, the lipolysis, and the biohydrogenation of PUFA. And if we look at the animal products, we could see that increasing the utilization of tannin increase the CLA in sheep milk, as compared to control. This is also the same message. Increase the PUFA, omega-3, increase the omega-6 as well, increasing the PUFA, alpha-linolenic acid, CLA. This is also not only applicable in milk, but also in meat. We could see here increasing the CLA in the meat of goat. This is also the same. Very interesting. So basically, the polyphenol increase the PUFA, including the CLA in animal products, in milk, and in meat. So it has, this is my final comment, this is Chairwoman. So apart from increasing the deposition of PUFA in animal products, tannin or polyphenol here has only a number of other beneficial effects. For example, it decreases methane emission. Decreasing methane emission from in vitro studies and from in vivo studies, both of them are consistent. Increasing also the animal performance. Our trial in a feedlot showed that the tannin-fed Brahman cross cattle increased. The average daily gain increased from 1.2 to 1.55 over 90 days period, one cycle of feedlot period. Why? This is because the protection of protein from the rumen degradation. So increase the bypass protein. Simultaneously, it also enhanced the healthy of gastrointestinal, by reducing the infestation of nematode. So it make the small intestine more healthy as compared to feed without any polyphenol. It also maintained the silage quality. So if we want to make silage, especially the high protein silage, like total mixed ration or also the alfalfa or other leguminous silage, it can inhibit the proteolysis So it reduces the ammonia, also reduce the butyrate, and it means that the tannin, the polyphenol, reduce the microbes in silage responsible for the deterioration of the silage. So I think I finished my presentation. If you have any question, feel free to ask me or we can discuss further. Thank you very much, Mrs. Chairwoman. Sorry for taking the time.
Thank you very much, Professor Dr. Anuraga, for the remarkable presentation. Okay, I will open Q&A session for Professor Anuraga. Please raise hand or type the question on chat forum if you would like to ask a question. While we waiting from the audience, maybe I will one question for you, Professor.
Yes, please, Ibu, please, very welcome.
Okay, in my opinion, as far as I know, lignin is also a polyphenol, but lignin and lignocellulose more than knowing as anti-nutrition. What do you think about that? We can also use lignin and lignocellulose to conduct high quality animal product or not, Professor?
Okay, thank you very much, Ibu. Very interesting question. Yes, actually, lignin itself is phenolic compounds as well. Yeah, but one of the different, it is deposited. If we look at the plant cell, in the plant cell, it is mainly deposited in the cell wall. So it enhances the rigidity of the plants. Yeah, and because of that main function, it enhances rigidity of the plant. Yeah, also against the infestation from microbes, from insects, and many other predators. So it is different function with the polyphenol. Polyphenol is synthesis by plant mainly, yeah, as the functional defense mechanism. So while lignin is the structural defense mechanism, but polyphenol is for functional defense mechanism. So mainly it interact, for example, with the enzymes, yeah, of the predators, so that they are not able to digest more the plants. And this polyphenol is mainly synthesis, yeah, in the plant during the young period of the plants, while lignin is increasing at the older stage. Yeah. So I think, and lignin in animal nutrition, yes, we are aware that lignin is negatively affect the animal performance. So if we increasing level of lignin, then it will reduce all the animal performance, milk production, meat production. So this is the other way around with the polyphenol. But polyphenol has also negative effects if we give it, if we give too much or too high concentration of the polyphenol in animal diet. So normally I recommend less than 1% of usage. Yeah, so less than 1% from the dry matter intake. So it will produce or increase the productivity of the animals. But when we use more than 2% or even more than 5% in dry matter intake, then it will reduce also the animal productivity. So our farmers is still consider the productivity as the main goal. More meat, more milk. They don't still today how to reduce the methane, how to increase the quality of animal products, yeah, like more unsaturated fatty acids. So we need to work on that one. Thank you, Ibu.
Okay, maybe another audience would like ask the question to Professor Anuraga. Okay, we have one question. Yes, in chat forum. From Mr. Tulus, there is any effect the modification to farm metabolism by dietary polyphenol in animal reproduction, maybe in male and female cow, like that?
Okay, animal reproduction, yeah, animal reproduction. So polyphenol, yeah, basically what importantly affect the animal reproduction is actually the antioxidant ability, the antioxidant property. So, for example, in the semen or for the males, it against the oxidation of the membrane of the semen or the cells of the sperm, the sperm of the male animals. So they are. I'm actually not concentrating on the animal reproduction, but I've read a number of papers that the use of polyphenol, it may also reduce the oxidation of the sperm. So basically, polyphenol has antioxidative property, and therefore can also protect the degradation or the oxidation of the sperm. So a number of plants containing polyphenols have been tested, and they are proven to increase the quality of sperm of the bulls or the animal or the ruminant males. But for females, I think most importantly it is PUFA itself, not the polyphenol. It's different logic. So if we deliver PUFA, high PUFA diet, and also the polyphenol at the same time, this can be beneficial also for the animal reproduction, I think, because this is the bioregeneration is limited or is decreased by the polyphenol, and at the same time it increased the unsaturated profiles to the sperm, for example, and at the same time also it enhanced the antioxidative property of the diet. I think that's all.
Okay. Unfortunately, our time has run out. I will.
Thank you very much, Ibu. I understand, really understand. Please do not hesitate.
Maybe the conclusion from Professor Anuraga, just short conclusion, maybe to conduct the high quality animal product, we can modify biohydrogenation of PUFA in ruminantia with polyphenol and PUFA combination dietary, maybe like that.
Right, Ibu, right.
Okay, we thank for the invite speaker for sharing the very useful knowledge this afternoon. Thank you.
Thank you very much, Ibu. Thank you very much, all audience.
Okay, so we continue the next agenda. So ladies and gentlemen, before we start the parallel session, maybe I would like to remind some rule of parallel session. First, all participant, presenter and non-presenter are expected to join as much as possible all sessions in this conference. Please use the virtual background provided by the committee. Please rename your Zoom username according to the following format. Please mute the microphone, especially during the presentation of keynote speaker. In the parallel session, presenter should activate the camera during their presentation. Questions from all participants are delivered using the raise hand, Q&A or chat feature in Zoom. When Q&A or chat feature selected, please deliver your question according to the format. Every session will be led by a moderator, and moderator will decide which question to be discussed or answered. All participants, presenter and non-presenter, should fill the list of attendees via the link given in each session. Attendees are not allowed to write opinion or information that is offensive, racist, or immoral in nature. Organizing committee have the full right to block and remove any attendee who break the above rule. That's our reminder. So we to the next session. Before that, Q&A in the first parallel session. maybe will be conducted after 3 presenter speeches. Okay, we move to the first presenter. We have Mrs. Wita Wardani with the title Phylogenetic Relationship of the Fern genus Atrium Athyracea from Indonesia. The time is yours.
Hello everyone. Assalamualaikum warahmatullahi wabarakatuh. My name is Wita Wardani. I'm a researcher in botany in the National Research and Innovation Agency. Currently, I'm also registered in doctoral program of Department of Biology, University of Indonesia. What I'm going to present here is part of my thesis work. Phylogenetic relationship of the fern genus Ecology from Indonesia. Atherium is a fern genus that included in the family Atheriaceae, formerly also known as in Woodsiaceae. This is often treated in broad concept that includes Symposium and Levaria. Current classification of Atherias is recognized as a Campium, Anopheles, Atherium, Symposium, and Levaria. And Atherium is part of the 3 most large genera in the family. It's a medium-sized industrial terrestrial fern. It has a rhizome with irregular ascending. The axis could be blackish or straminious. It has axes that also come from V-shaped groove. The stipe scales are not quadrate. It has an entire margin and rarely hairy. Sorry around reniform, or J-shaped, linear oblong, and it's induciate. Main distribution of this genus is in the Eastern Asia. As also other large taxa, Atherium has disagreement of species delimitation and perplexing relationships. There is a global phylogeny for Atherium, but Indonesian species are rarely represented in that analysis. Therefore, the aim of this work is to evaluate phylogenetic position of Indonesian Atherium species within the global phylogeny. So we have 54 accession of 41 species, that 39 is in-group and 2 are all-group. Among the 54, 39 are newly generated. So DNA extracted from silica dry material and amplification on chloroplast DNA, 3 chloroplast DNA that one consists of 1 coding region and 2 non-coding regions. Phylogenetic reconstruction was based on molecular datasets using methods of maximum parsimony, maximum likelihood, and Bayesian inference. For maximum parsimony, we carried out using the software of Mega, while for the maximum likelihood and Bayesian inference, the analysis performed on the cytoscience gateway using Raxanel and Mr. Bayes. The concatenated DNA sequence contains 953 variable sites of the 2,503 base pair aligned sequences. The tree analysis generate trees with similar topology. The newly generated sample from Java are nested in the section of Photophora and section Foresticoides. The brown and the orange boxes, respectively. In the global anterior phylogeny, there is an unexpected relationship with Bornean and Chinese species. On your right side is the maximum parsimony 3 topology, and this one on the left side is the maximum likelihood 3, and the right-hand side is the Bayesian inference. All 3 have similar topology. The the Japanese species of Asterium licopes are nested in the tip of the clade section of Topua. That surprisingly, a Bornean species, Asterium amoenum, also nested in that subclade, in the same subclade. On the contrary, Asterium licopes that identified from China were nested in different clades with the Japanese species. On the other hand, the pinate form Asplenium gedeanum were in close relationship with Asterium erythropodum, which has a very different leaf dissection, while with the similar looking species of China, Asterium caesalpinum, were nested in different clades with Asterium gedeanum. So our findings show that based on the three chloroplast DNA markers, Japanese species Asterium nicotus nest at the tip of the clade of Photophora, with the Bornean species included in the same subclade, which this merit further clarification for synonym, while similarity in morphological appearance does not in agreement with phylogenetic analysis, that demonstrated in the relationship of Asterium gedeanum and Asterium caesalpinum that separated in different grades. This also merits a further investigation in that deeply into character evolution. Thank you for listening.
Okay, thank you, Mrs. Wita Wardani. Next, we to the second presenter is Mrs. Ruliana Susanti with the title Tree Diversity in the Buffer Zone of Bukit Baka Bukit Raya National Park. The time is yours.
Good morning, good afternoon. In this opportunity, I would like to present our research result entitled Tree Diversity in the Buffer Zone of Bukit Baka Bukit Raya National Park. For the introductions, IPB's global assessment already highlights some examples of declines in nature. For example, 47% natural ecosystems are declined and 25% of species are already under extinction. A transformative strategy is needed to halt the declining process, including good management of protected area. Bukit Baka Bukit Raya National Park is one of the remaining in situ biodiversity conservations in the heart of Borneo that plays an important role in strengthening biodiversity conservation, ecosystem management, and services. The forest cover from 2012 to 2017 is not significantly changed. However, its biodiversity potential and threat is still need to be revealed. Higher threat is faces especially in the areas that border with the human settlement. Thus, this research objective is to determine plant diversity and its conservation status in the buffer zone of national park. For the methodology, the study site is in Bukit Baka Bukit Raya National Park located in two provinces, Central and West Kalimantan, as we can show in this picture. The national park covers more than 200,000 hectares of forest and hosts large rivers, important as water source for both provinces. Survey was conducted in the border of national park with the locations of PT Saribumi Kusuma, a forest company concessions. So the national park is in the right side and the company is in the left side. A transect plot size 10 by 50 meter squares were established in 4 locations between 100 to 500 meter above sea level. All 3 were with diameter more than 10 centimeter were measured, collected, and preserved for further identifications. Tree diversity, structure, and compositions. Also, conservation status is analyzed. The normalized difference vegetation index analysis is conducted to evaluate forest cover change. All data were then analyzed using R with Pagan and GG2 package. For the research result for the forest characteristic, our study are able to collect 352 species, tree samples from 35 family, 58 genera, and 159 species. The Simpson Diversity Index analysis shows that among 4 locations, tree diversity is quite even though in terms of basal area, the dominant species is quite different in the 4 locations. For example, in location 1, Tristania, Syzygium, and Soreolithus dominate the area, and location 2 dominated by Sorea multiflora, Compassia malacensis, and Sorea virescens. Location 3 dominated by Sorea dasyphylla and Sanctophyllum, while location 4 dominated by Triobalanops and Compassia malacensis. However, if we look up to the family level, we can conclude that Dipterocarpaceae is the most diverse family in terms of species number, followed by Phyllantaceae, Myrtaceae, Anonaceae, and Mysticaceae. Next, Dipterocarpaceae not only dominated in terms of the number of species or the species richness, but also in terms of density, it covers 22% of the whole tree diversity, or the tree collected, and in terms of basal area, it covers 45% of the total basal area. For the diameter distributions, most of the tree found inside the plot has diameter less than 60 centimeter, including also for the Dipterocarpaceae species, and only 3 individuals found with the diameter up over 100 species. Two of the 3 individuals is Soreolithus and Sorea multiflora, as you can see in this picture. The Soreolithus at the left picture and Sorea multiflora at the right picture. Both of the species has a very similar leaf shape, so it is difficult to identify actually for Dipterocarpaceae up to species level. So there is possibility that if the sample of the reproductive organ can be collected, thus the number of species can be increased than that we already collected and identified. We are able to identify 28 species of Dipterocarpaceae for 4 genera: Dipterocarpus, Driobalanops, Hopea, and Sorea. And Sorea has the most diverse species compared with the other genera. Among the 28 species, 16 species is under threatened status of IUCN, as you can see in this table, and 12 species is endemic to Borneo according to the most recent publication by Bartolo et al. 2021, of which 8 species is under threatened according to IUCN. There are 1 species in the critical endangered, 1 endangered, and 6 under the vulnerable status. The critical endangered species found is Hopea mangerawan and Sorea rotundifolia, while the endangered species is Sorea dasyphila, Sorea pauciflora, Sorea splendida, and Sorea sumatrana. Sorea rotundifolia and Sorea splendida, despite its threatened status, it also has an endemic status for Borneo. This picture shows the pictures of Sorea rotundifolia. According to the most recent publications in Borneo, 11% of the Dipterocarpaceae is under the critical endangered status, and 25% is under endangered status. Thus, it is very important to develop the conservation strategy for the Dipterocarpaceae in this national park. For the forest change, the NDPI that we use is commonly used to observe the land cover change in an area. As we can see in this graph, apart from the optical and atmospheric problem of NDPI and Landsat 7, the value of pink indicate that the conditions of the forest is very stable and quite good in the last 10 years. This is also supported the previous literature that also state that the forest cover change in this national park is not too big. For the conclusions, forests at the border of the national park with the company concession remaining in quite stable condition for the last 10 years, and the diversity is quite high with 139 species. The forest border was dominated with the tree from the Dipterocarpaceae in terms of species richness, density, and basal area. Among 28 Dipterocarpaceae species, 16 species can be considered as threatened according to IUCN assessment, and there are 12 Bornean endemic Dipterocarpaceae, of which one species is in critical endanger and one species under the endangered status. This is the end of my presentations. Thank you very much for your attention. This research is funded by the Strategic Fund for the Promotion of Science and Technology of Japan, or GST, collaborations between LIPI and Kyoto University and facilitating by PTSDK. Thank you very much.
Thank you, Mrs. Juliana, for the interesting presentation. Okay, next we have the third presenter is Mrs. Nikade Erosi Udaharta with the title Population, Genetic Diversity and Structure of Endangered Species, Dipterocarpus hasselti bloom, using genome-wide RAD sequence data in Bali, Indonesia. Okay, the time is yours.
I'm Nikade Erosi Udaharta from Research Center for Plant Conservation. I'm going to talking about population genetic diversity and structure of endangered species Dipterocarpus hasselti using genome-wide RAD-seq data in Bali, Indonesia. Dipterocarpus species dominate lowland Southeast Asian tropical forests, and most of the 14 genera occur in the Malaysia region, with some representative in Africa and South America, but of far less species. The Sundaland rainforests are dominated by Dipterocarps, and there is striking distinction between Asian Dipterocarps and those of the African American tropic is most likely due to Sundaland's long isolation from other tropical regions since the Middle Cretaceous period, and obviously distribution function of Dipterocarp forests as an indicator for the extent of the rainforest in Sundaland, and as the center of Dipterocarp diversity center and dominate from Dipterocarpaceae family. And as we know that Indonesia has a forest area of 92 million hectares, which makes up 2% of the world forest, and Indonesia is one of the 17 megadiverse country in the world, and moreover, Indonesia contains area of high species endemism that are at risk of habitat loss and loss of genetic diversity. And nevertheless, the high level of exploitation and degradation in Indonesia led to many species being threatened. Deforestation and land use change diminishes the remaining habitats of many threatened tree species. However, secret groves rose in prominence as a refuge beyond the protected area network, and secret groves located within a human-modified landscape often act as a node of resilience for threatened tree species, and especially when the tree species is experiencing a large-scale habitat destruction, the tree species has high economic value, and the tree species has cultural, religious importance. And we know that secret groves are forest species conserved on the basis of religious beliefs and cultural practice of local communities. And dipterocarps are important source of timber market in global economy, such as Dipterocarpus hasseltii. And we know that Indonesia, as one of the world's largest exporter of tropical timber product loads, however, habitat fragmentation reduces the population size of many Dipterocarps species and their survival. Moreover, forest degradation causes biodiversity and endangered species to be threatened in nature. And to support sustainable forest management, it is necessary to assess the ecological value and the genetic value of endangered species. And therefore, in order to preserve endangered species from extinction, it is important and crucial to study their population genomic through conservation genetic concepts. And knowledge of genetic diversity and population differentiation of Dipterocarpus hasseltii are important for its conservation. The use of Dipterocarpus hasseltii not only could prevent murine leukemia, but in Bali, Dipterocarpus hasseltii by local people as an offering result, and this has resulted in the widespread distribution of this species on this island. Due to the importance of Dipterocarpus hasseltii as a timber tree and part of religious offerings, higher levels of legal protection and scientific research into the species should be strongly encouraged. And we know Dipterocarps are long-lived trees with gravity dispersed seed and high genetic diversity with a strong genetic structure. And however, fragmented habitat had negligible effect on genetic diversity. And however, high outcrossing rate 80% and high tendency of mating system between relatives. This is a clear genetic structure in areas smaller than Bali, and the important thing is no population genetic studies on trees in secret groves. A total 106 leaf samples from protected areas, forests, and secret groves in 8 populations were utilized for the present study, and genomic DNA was extracted from fresh leaves sample using the plant genomic DNA kit and using the modified CTAP method, and the purified DNA was sequenced pair end sequencing 2 until 200 base pair, and to until 3 GB per sample using Illumina HiSig 4000 platform. And I'm using Stack for quality control and snip calling and analyze data in R. And the meeting happened more often between individuals or who are more closely related than between individuals taken at random from the subpopulation, and lower genetic diversity typical of isolated population at range age. And the A population showed low level in breedings. And for the specific finding revealed that only a few individuals have mixed descent from more than one genetic cluster. And therefore, a small isolated population of Dipterocarpus hasseltii have slightly low genetic diversity, and it is clear there is no genetic erosion among population in Bali. PCOA was used to investigate the genetic relationship among Dipterocarpus hasseltii individuals, and the findings were consistent with the structure analysis as distributed throughout the entire genome. And the population show two clear groups, and other groups are indicated as mixture-aged individuals. And geographical distance among this population could be a factor driving the high genetic differentiation observed here. And for example, NP and YE were divided by a large distance of more than 60 kilometers. For the conclusion, are no evidence of genetic erosion, and geographical factor had a strong influence on gene flow and limited source for all secret group population. And although low level of genetic diversity, nevertheless, all Dipterocarpus hasseltii population should be conserved. And threatened species and isolated population are main reason to consider as priority to conserve of Dipterocarpus hasseltii in Bali. And for the best management approach for this species is to develop an ex situ conservation site using seed harvested from all population of Dipterocarpus hasseltii, preventing the spontaneous loss of genetic diversity caused by the loss of individual ecotypes and maximizing the survival of new self-sustaining population. Okay, that's all. Thank you.
Okay, thank you to Ms. Nikade Erosi. So now we in a Q&A session for three presenters. Please raise hand or type the question on chat forum if you would like to ask a question to one of the presenter. We already have one question in chat forum from Bu Wita Wardani to Mrs. Ruliana. Is there any comparison from the other part of national park on its diversity with current management practice? Do you think that particular border site where your research was conducted will be stable for longer term? Okay, Ms. Ruliana, you can directly answer this question.
Hello, can you hear my voice? Can you hear my voice?
Yes, I hear.
Oh, thank you. Thank you for the questions. So to answer the questions, first, yes, there are several other plots in the national park, but it's not located in the border area. And I'm sorry that I didn't put it in the result part of this of the presentations. In the other area of the national park, the diversity also quite similar the index is quite similar with with the diversity index that we found in the border area. However, since the methodology is a bit different, so we cannot directly compare the result. Then for the second questions, with the current management practice, I think yes, the border site will be quite stable for long term since the location is difficult to access. The location is quite close to the high conservation value of the company. So it's quite difficult to access. So the local people or common people cannot directly enter the location. So I think it will be quite stable for a long time. Thank you.
Okay. Thank you for the great answer from Ms. Ruliana. Okay, maybe from the other audience, there is a question for the other two presenters, maybe. Okay, while waiting the audience, I have one question for Mrs. Wita, because I a chemist, not a biologist, so I just curiosity about why the relationship with Borneo and China species from your atrium conducted the unexpected result from your presentation. Actually, what are the expected result? Truly, maybe like that, Ms. Vita.
Thank you for the question. So yes, it was quite surprising that the Bornean species Atherium amoenum was identified by the expert. The sample is from the Kinabalu, so it's quite far from my sample that came from Java. But however, with
Kinabalu, where is Kinabalu?
In Borneo, in
Borneo.
Malaysia, and
Malaysia.
In Sabah and Sarawak.
Oh.
So fern distributed by wind. So it really possible that some species distributed very widely, and we often think that something occur in Kinabalu is restricted, isolated from everywhere. So it's really endemic to Kinabalu. But then my result sort of confirming that Kinabalu might be not that unique anyway. So there's a possibility whether the person who used that samples, because that's not my sample, it's somebody else, probably he was misidentified. It should be nigripes, but then he identified it as amoenum. But there's a chance as well that it's actually the same species. So amoenum is a synonym for nigripes. So then the next thing is the nomenclatural aspect that need to be addressed. But without access to the sample that being used, I cannot really tell whether which one is the correct one. So I really want to go to Kinabalu and find that plant itself.
Okay.
Thank you for the question.
Okay, thank you, Mrs. Wita. Okay, for the last question, maybe we go to Ms. Nikade. This question from Muhtar Davis from BRIN. He asked, you did population structure analysis on dipterocarp Hasselti by collected DNA sample of individual throughout Bali and perform genotyping. How did you ensure that you collect specimen from dipterocarp Hasselti and not other dipterocarp species? Okay, Ms. Kade.
Okay, yeah. Thank you so much for the question, Mr. Mukdar Davis. Yeah, for my samples from the Dipterocarpus hazeltia, I take for some specimen, such as from the secret groups and protected forest, which is the leaf samples I collect and I helped from the taxonomists who knows about the Dipterocarpus hazeltii, and then before I take for the leaf samples for the collect, I asking about that. Even for the seedling, I just check carefully in the field, and then after that, I take for the leaf samples and then collect. And then doing for the DNA extraction, like that. It's just make sure about the Dipterocarpus hazeltia is right, and then I'm not using some taxonomists help me to identify which one is Dipterocarpus hazeltia in the field. Thank you.
Okay, thank you.
Yeah.
Thank you for the great answer from the three presenters, and we in 2 p.m. and 40. So our time have ran out. So I thank you for your attention, and I apologize if there are mistakes during this session. Next, we will have a video session, and will be continued by Mr. Dimas Fandi Praditya, Magister of Biotechnology, as moderator on second parallel session. So I conduct the next session to committee. Thank you.
Thank you very much, Dr. Evi Susanti. Ladies and gentlemen, before we continue to the parallel session 2, we will see a video session from our sponsor.
Berikut ini adalah video yang menampilkan proses pengerjaan sampel dengan menggunakan QIAcuity Digital PCR. Seperti yang telah kita ketahui, QIAcuity Digital PCR didesain untuk mengerjakan sampel dengan mudah, cepat, dan kapasitas high throughput. Untuk mengerjakan sampel dengan QIAcuity, kita membutuhkan QIAcuity PCR Kit, nanoplate, instrumen, dan QIAcuity Software Suite yang sudah terinstall pada software atau laptop yang kita gunakan. Pemilihan PCR Kit yang digunakan disesuaikan dengan aplikasi. Terdapat dua jenis reagen QIAcuity Digital PCR Kit, yaitu QIAcuity Probe PCR Kit dan QIAcuity Eva Green PCR Kit. Selain itu, kita juga perlu mempersiapkan peralatan standar laboratorium serta nanoplate.
Terdapat 3 tipe instrumen QIAcuity, yaitu QIAcuity One untuk 1 nanoplate yang memiliki pilihan deteksi 2 fluorensi dan 5 fluorensi, QIAcuity 4 untuk 4 nanoplate yang dapat mendeteksi 5 fluorensi, dan QIAcuity 8 untuk 8 nanoplate yang dapat mendeteksi 5 fluorensi. Hanya QIAcuity One yang tersedia dalam pilihan 2 plex. QIAcuity memiliki 3 modul pengerjaan, yaitu priming, proses membuat sampel masuk ke dalam partisi dalam nanoplate, kemudian thermocycling modul untuk proses amplifikasi molekul target, dan yang terakhir imaging modul, yaitu pembacaan sinyal fluorescence.
Berikut ini saya akan menunjukkan tipe nanoplate yang tersedia. Di sini sudah tersedia 2 tipe nanoplate, yaitu nanoplate dengan 8.500 partisi, 24 well atau untuk 24 sampel. Dan tipe yang kedua adalah nanoplate 8.500 partisi untuk 96 sampel. Untuk pemilihan dari tipe nanoplate yang akan kita gunakan, itu nanti akan disesuaikan dengan aplikasi eksperimen yang akan dikerjakan. Di sini pada bagian belakang plate kita dapat melihat terdapat kotak-kotak persegi. Ini adalah bagian dari partisi yang terdapat di dalam nanoplate. Jadi ketika kita mengambil nanoplate dari box, itu pastikan kalau kita hanya memegang bagian pinggirnya saja. Jangan sampai terpegang bagian dari belakang. Kemudian ketika kita untuk men-set nanoplate, pastikan kita menggunakan tray yang sudah disediakan pada saat kita melakukan pembelian dari nanoplate.
QIAxcel Advanced, an introduction to fully automated capillary electrophoresis. This is the QIAxcel Advanced. QIAxcel Advanced uses capillary electrophoresis for effortless DNA and RNA analysis. Capillary electrophoresis separates analytes within a gel when an electric field is applied. To run QIAxcel Advanced, you will need QIAxcel cartridges that are specific for your applications and the QX nitrogen cylinder. You can analyze between 12 and 96 samples per run. At the top of the QIAxcel Advanced, users have access to control the nitrogen pressure and also to exchange the purge filter in case it needs to be replaced. Insert the QIAxcel cartridge and the smart key. Load the samples and buffer tray onto the work table. Ensure that the QX nitrogen cylinder is fully tightened. Alternatively, you can connect an external nitrogen supply. The instrument requires nitrogen to apply the required system pressure. At the rear of the instrument, the nitrogen port can be used to supply external nitrogen. Load your samples, markers, and buffers. The Smart Key contains information about the cartridge ID, the number of available sample runs, and calibration runs that have been performed, the successful calibration with the QIAxcel Advanced, and the cartridge expiry date. The gel reservoir of the cartridge contains the gel which is used for the run. The gel is replaced for each new run. Nitrogen pressure is applied via the purge port, enabling the instrument to replace the used gel from the previous run with fresh gel for the next run. Analytes are loaded into the gel capillaries and separated by size under the application of an electric field. Unused cartridges should always be stored upright at 2 to 8 degrees Celsius in the packaging blister or in the covered cartridge stand to protect the gel in the cartridge from drying and from direct light. If the cartridge is stored in the KayakCell Advanced, the instrument should always be switched on. Before using the cartridge, it is crucial to equilibrate the cartridge for at least 20 minutes to room temperature to ensure proper analyte separation. To equilibrate the cartridge, either place it into the cartridge stand, including wash buffer overlaid with mineral oil, or directly place the cartridge into the KayakCell Advanced. Ensure that the buffer tray is loaded and wait. Incomplete temperature equilibration can cause run problems and potentially also lead to instrument damage. Always use the correct cartridge type together with the appropriate buffers and markers. Thank you for watching this video. We hope you find it useful.
QIAGEN. Sample to insight.
To all participants, let's continue to the session 2 of this session. And this parallel session will be led by Dr. Dimas Mandipraditya from Research Center for Biotechnology, Jodha. Dr. Dimas, the is yours.
Okay, good afternoon, everyone. First of all, I would like to say thank you to organizing committee that allowed me to lead this parallel session, and also for the all participants that already choose this room for the afternoon session. I hope you all still awake. And before I start this session, I just want to remind you about the general rules. So please turn off your microphone while the speaker is speaking. And if you have a question, you can just raise your hand or you can just write in the chat box. And please address to whom the question is. And we will start discussion after the last speaker is finished with his presentation. And in this session, we will have a 5 speaker. First, Ibu Dwi Ningsih Susilowati from Indonesian Center for Agricultural Biotechnology and Genetic Resources, Research and Development. And then Bapak Sutomo from Research Center for Plant Conservation, BRIN. Next, Ibu Senli Octaviana from Research Center for Biotechnology, BRIN. Bapak Suyadi from Research Center for Biology, BRIN. And I hope I correct, this is Ibu Sarwa Handayani from Center for Research and Development for Biomedical and Basic Technology, National Institute of Health Research and Development, Ministry of Health. So now we can start for the first presenter, who is Dwi Ningsih Susilowati. The time is yours.
Thank you to moderator. I am Dwi Ningsih Susilowati from BBBU again. Today I would like to present my research with title Fungal Community Analysis in Acid Sulfate Soil from 2 types of swampland in Kalimantan using next generation sequencing. Research background. Acid sulfate land in Indonesia cover approximately 6.7 million hectares and 1.7 million hectares in Kalimantan and others in Sumatra, Sulawesi, and Papua. Acid sulfate soil are divided into 2 types, that is actual and potential acid sulfate soil. In the study, acid sulfate soil in South Kalimantan are classified as potential type B because this land only overflowed by large tides, which usually occurs during the full moon. While acid sulfate soil in Central Kalimantan belong to actual type C, where this land is not flooded by large or small tides, but the depth of groundwater range from 0 till 50 centimeters of the ground surface. Acid sulfate land has the potential to be developed for agricultural land to increase food production in Indonesia. But this land have not been utilized optimally because of soil productivity is very low due to high soil acidity and soil nutrient deficiency due to high level of aluminum and iron. Therefore, it needs special handling so that acid sulfate land can be developed as productive agricultural land. Technological innovation, especially fertilization, it must be pay attention to the indigenous microbial community in the soil. So the application of biological fertilizer technology can be effective. In this research, we focus on fungal communities by using next generation sequencing technique to study fungal community in two types of acid sulfate soil. The objective of my research is analyzing the diversity and composition of fungal species on acid sulfate soil from potential type B in South Kalimantan and actual type C in Central Kalimantan, and knowing the level of acid sulfate soil fertility in two types of swampland. Research methods. Soil sampling. The soil was sampled from two types of acid sulfate land. One potential type B from South Kalimantan, exactly obtained from Tanjung Harapan village, Alalak subdistrict, Barito Kuala Regency, South Kalimantan province, and actual type C from Central Kalimantan, exactly obtained from Tamban Baru Tengah village, Tamban Catur subdistrict, Kuala Kapuas Regency, Central Kalimantan province. And then the soil sample from two locations were analyzed for the physical, chemical, biology properties, and after that we carried out microbial or fungal community analysis by using next generation sequencing and continued with data analysis. Result and discussion. Table 1 showed soil physical properties of acid sulfate soil from two locations. Soil from South Kalimantan with potential type B as a silty clay or dusty clay, while soil from Central Kalimantan with actual type C has a clay loam texture. It does have low water holding capacity but easier distribution of nutrient. So soil type potential B in South Kalimantan is more better than actual type C soil in Central Kalimantan. Table 2 showed soil chemical properties of acid sulfate soil from two locations. Two types of acid sulfate soil showed a very acidic soil with pH less than 4.5. The CN ratio of acid sulfate soil from two locations are classified high, which can cause immobilization of nutrient and accumulate organic matter so that it inhibited the process of decomposition. How about the kalium and phosphorus content? Both of potential and actual type have low kalium content, which is associated by the high acidity of the soil pH, but the phosphorus content are classified high. And aluminum content was classified as very high. The aluminum is toxic to various crops in swampland. This picture showed the result of total plate count of fungi grown on rose bengal media from both acid sulfate soil samples. We can see that fungal population in potential type B South Kalimantan was higher than actual type C Central Kalimantan, both at normal pH and acid pH. This is, of course, related to the physical and chemical condition of the potential type B South Kalimantan, which has a relatively better than actual type C Central Kalimantan. Fungal diversity into location based on the operational taxonomy unit. From this picture, we can see that the number of OTU obtained in South Kalimantan soil is more higher than Central Kalimantan soil. These results are in line with the result from culturing on rose bengal media. The result of the number of OTU obtained in South Kalimantan are 390 and Central Kalimantan 399, with a total number of OTU from both locations in 719. And what kind of phylum and genera are dominant? From this picture, we can see that Ascomycota is a phylum that dominates both types of acid sulfate soil in Kalimantan. And a total of 78 genera were identified from both locations, with 28 unique genera in South Kalimantan and 23 unique genera in Central Kalimantan. Table 3 showed alpha diversity index of Chow richness, Simpson diversity, observed species, and phylogenetic diversity. Both of potential type B and actual type C have the Simpson diversity index is close to 1. It indicated that the composition of the community is more complex. Chow1 is a non-parametric index that estimates species richness based on UTU. The results show that the Chow1 value at South Kalimantan 385, while at Central Kalimantan it's lower at 324. Analysis alpha diversity index in table 3 showed the species identification and potential type B South Kalimantan amount 385 and in actual type C amount to 310. Venn diagram showed the similarities and differences of species between two locations. South Kalimantan and Central Kalimantan have the same 207 species with a total of 178 species that are only identified in South Kalimantan and 112 species of which only in Central Kalimantan. The conclusion of my research is fungal diversity and abundance of acid sulfate soil in South Kalimantan, potential type B, and Central Kalimantan, actual type C, obtained from the NGS analysis consisted of 5 phyla, 16 classes, 40 order, 69 families, and 79 genera. Acid sulfate soil in South Kalimantan with potential type B has a better fertility level than Central Kalimantan, actual type C, seen from the analysis of soil physical, chemical, total plant count of soil fungi, as well as the diversity and abundance of fungi from both locations. That is all my presentation. Thank you for your attention.
Okay, thank you for the nice presentation, Dwi Ningsih Susilawati. Now we move on to the next speaker, which is Bapak Sutomo from Research Center for Plant Conservation, BRAIN. The time is yours.
Hello everyone, thanks for your attention to our presentation about habitat suitability modeling for Jalak Bali, Leucopteros silvi in East Java, Bali, and Lombok. A potential set for its exit to conservation. Our team are Sutomo, Luputu Eswaryanti Kusuma Yuni, it's me Rajiv Wiryadi, and Eddie Van Itten. To introduction, we know that Bali starling is endemic species of Bali Island. It has critically endangered with small population size, restricted to a small area illegal trapping, and the suitable habitat within its natural range was decreased. This record 21 individual in 2015 in Bali Barat National Park. From historical release in situ area in June 2008 and June 2009, 20 individual to release, and October 2010 have 22 individual, and there are have a problem was illegal trapping. And next, the information from exit to area in several Bali in 2019 have 14 individuals to release to nature. This study objective is to apply spatial modeling technique to find the suitable habitat of Bali starling. Next, potential set for exit to conservation having minimum effort. The method. The first step, we have identifying the general spatial distribution We use spatial distribution modeling with BCCVL, with generate of the spatial distribution of this species with data occurrence from GBAF and environmental data from world current condition. These variables such as precipitation of trace amount, annual mean temperature, isothermality, annual precipitation, temperature seasonality, precipitation seasonality, and precipitation of wetness for month. Next step, we get a potential set for ex situ conservation. With the spatial distribution modeling result, adding information with GIS analysis with native environmental detail, such as isothermality, rain temp, elevation, and land cover to have the result. And the result, we know trend pattern existing Lake Abstaros Sildi in 1974 to 2014 have a positive correlation between number of habitat spotting and amount of total population. We detail from the graph in this presentation. And next, highlight several places that have similar habitat suitability index with the original habitat of Bali starring in Bali Barat National Park. Bali starring prefer lowland area, preferably occur in less dense forest, secondary forest, monsoon or dry forest, and with adjustment savanna or grassland or more open vegetation. Identifying similar condition, we have obtained from the variable with the BCCVL analysis is the significance from the variable B3 and B12. In B3 is optimum rate of isothermality index in 0.84 to 0.86. But we use the temperature range data on native local cost, the isothermality index linear to temperature. And next, the optimal annual precipitation habitat is 1,000 to 1,800 millimeter per year. This detailing information, we have 4 variable to detailing the data BCCVL or the species distribution model. This as the variable the range of temperature such as the isothermality, and the rain classification is the distribution from the in situ location in Bali Barat National Park is the based on there. And next, like cover classification is the based on the forest area and non-forest area. In the elevation, we get the elevation less than 500 meter from ISL. And the result shown that is suitable at Baluran, Banyuwangi, Alas Purwo, the whole of Bali, including Nusa Penida, expert highland area. And the Lombok is spare from the Lombok area, expert highland area too. And from conclusion, spatial analysis combining biotic and environmental data helps information about the potential of the area of ex situ conservation of the Bali starling that has limitation in manipulation environmental variable. Bali starling prefer lowland area, preferably occur in less dense forest, secondary forest, monsoon or dry forest, and with adjacent savanna or more open vegetation where are at Baluran, Banyuwangi, Alas Purwo, the whole Bali expert highland, and southwest of Lombok Island expert highland area. Okay, thanks for Bali Barat National Park Management, Mr. Wiryawan, to supplying the distribution data of Bali starling and the website modeling platform BCCVL. Okay, thank you.
Okay, thank you for Bapak Sutomo. And we shall go to the next speaker, which is Dr. Senli Octaviana from Research Center for Biotechnology Brain. The time is yours. Maybe we can...
Hello everyone, my name is Senli Octaviana. I came from RC Biotechnology Brain. Today, I will give you an overview one of my project entitled Evaluation of Multilocus Sequence Analysis for the Phylogeny of Myxobacteria Isolated from Indonesian Mangrove. This project has collaboration with Dr. Chandrawati Moses from RC Chemistry, also from BRIN, and PD Dr. Joachim Wing from HZI Germany. In this presentation, I divided into 5 parts. Let's start from introduction. So, do you know Myxobacteria? Myxobacteria is one of member Delta Proteobacteria that have a unique social behavior because they moving, preying, killing, and surviving together. You can see here that this is one example of Myxobacteria. This is Myxococcus xanthus. They prey to E. coli, and when the nutrient limit in their environment, they convert to fruiting body. So you can see here that this is the dot is a fruiting body, and that's why I said before that they surviving together. When the environment become rich again, they start to prey, or we can say, or kill to other bacteria. So in the right side, you can see the kind of the fruiting body here. In the right side, this is one example of rare Myxobacteria, namely chondromyces. You can see here that this is a beautiful Myxobacteria. They make like a tree. Myxobacteria, also known as microbial natural product, because they produce bioactive compound. For example, this is ambrositin from Sorangium. Ambrositin is for the first bioactive compound that isolated from Myxobacteria. Taxonomically, until 2019, more review this Myxobacteria. Myxobacteria consists of 3 suborders. First is Cystobacterinae, Soranginae, and then Nanocystinae. And then they divided into 11 family, 29 genera, and 66 species. Today we talk more specific in Myxococcaceae family, specific to Myxococcus species. So the background of my research is based on the previous study that I isolate the Myxobacteria from Indonesian mangrove, that the Myxococcus species is abundant in there. So I selected 5 Myxococcus based on their activity, because I mean, based on the activity, they have a different character. For example, here that accrued extract of 4 to 1 MSO against Specia anomala, but they have a 33.33 microgram per milliliter. It means it's different with other, but when I identified based on the 16S rRNA, they belongs to Myxococcus macrosporus. The next question is there any tool to make the differentiate between strain? Here, we know that the 16S rRNA gene is still fundamental of the identification, and that's why in this research we try to see how MLSA approach to identification and classification of the 5 Myxobacteria strain. So the method, so MLSA is a phylogenetic analysis of multiple internal fragment of gene. It's mean from housekeeping gene. Here that the MLSA, we choose for the first, we choose the housekeeping gene here, and then we make a concatenate, and then we reconstruct the phylogenetic tree, and then we compare to the 16S. After we got the DNA from 5 mixostrands, of course, first we make sure for the identification of this strand using the 16S RNA gene that I mentioned before. And in this study, I used the 4 housekeeping gene consists of GIRB, PIRG, RPOB, and also PGM. For the result, this is the table, 5 isolate with the accession number of each housekeeping gene and also the 16S. But you can see here when we see the PGM gene, the strand in 191 MSO, there is no information because we omitted, even we amplified this gene with the in PCR, it's good, but when we try to sequence, it's really bad, and it's happened like several times. That's why we choose or just omit this gene to strand 191. So based on the phylogenetic tree here, when we compare the 16S and concatenate, first, this is the concatenate for housekeeping gene without 191 MSO. You can see here, for example, we can define each strand. For example, the strand 421 close together with the 471 make a one plate, and it can differentiate with other strand. Also, same result when we use the concatenate with 3 housekeeping gene, that we can divide it into several brands. It means when we try to make a similarity of this concatenate with the 16S, you can see here the black one is for the intraspecies and the red one is the interspecies. With the 16S, the similarity above 98, but when we use the concatenate, you can see here that the both of intra and interspecies is average under 98. It means that we can differentiate it between strand or between a species. So the conclusion is even the 16S fundamental of for identification of the bacteria, but we can use the alternative tool or method with the MLSA to distinguish strain. And this is we can say that this is an alternative tool for the phylogenetic tree. So thank you very much for DAD for financial support for this project, and also Joachim member here for supporting my work, and thank you for your attention. If you have a question, don't hesitate contact me.
Okay, thank you, Dr. Senli Octaviana. Actually, this is interesting subject, and I have a question, but we can discuss after the last presenter. So now we can move on to the next speaker, which is Bapak Suyadi from Research Center for Biology, BRIN. The time is yours.
Hi everyone. Today I'm going to present our research. The title is Battery Approaches Are Required for Successful Mangrove Restoration and Rehabilitation Program. Here we can see from the map that most of mangrove in the world located in the tropical region, including Indonesia. You can see here tropical mangrove, and then this is the temperate mangrove. Tropical mangrove cover more than 99.6% of the global mangrove. You can see here that Indonesia is the archipelago countries, and we have a lot of small islands, and our coastline is the second longest in the world. So this is a good habitat for mangrove forests. So we can see here that Indonesia is the largest mangrove habitat, almost 23% of the mangrove located in Indonesia. Not only that, but also Indonesia has species biodiversity. For example, 97% of global mangrove species are located in Indonesia. And 14 species of mangrove is endangered species, and 3 species are endemic species. And mangrove also provide a lot of ecosystem services. For example, here we can see that carbon sequestration or carbon stock, both above ground and below ground carbon stock. Mangrove also a habitat for many benthic and also birds, for example. And this also have a function for shoreline protection and also filter runoff. People around the forest or in the coastal area also use mangrove as a raw material of many kind of foods, drink, or anything they need. And however, we have a big challenge because 60% of global mangrove has been lost in the last 16 years, 2000 and 2016. And we can see here in Indonesia, mangrove decreased significantly. Almost 45% of mangrove in Indonesia is degraded or deforested. This is because of mostly due to the aquaculture. Here we can see here is shrimp pond, and also because of the illegal loggings and also plastic pollutions. It's the big problem of mangrove ecosystem in Indonesia. All of the threats of mangrove ecosystem are anthropogenic pressure on the mangrove ecosystem, and the most greater threat is mangrove deforestation and degradation. So this way, many communities and the government and also companies also try to restore or to conserve the mangrove. 2 things, 2 conservation action conducted mostly is conserving the remaining forests and also restore the degraded mangrove or dead planting mangrove. But we need to evaluate mangrove planting. Is this in the right place? Is it successful or not? So this evaluation is important to support the mangrove restoration program. For example, currently the Indonesian government, they just established a new agency, what we call Badan Restorasi Gambut and Mangrove. And this evaluation is also important to better management of mangroves. Our study site is across Indonesia, mainly in the eastern part of Indonesia. You can see here we have 8 study sites from Segaranakan in Central Java, Weda Bay, Ambon Bay, K Island, Seram Timur, Cikumerasa, Bentenan, and more islands. And all of this is the habitat of mangrove represented in small and big island. We applied 3 main methods: vegetation plots and questionnaire survey, followed by interview survey to clarify that the question is correct, or to clarify if the answer is not clear. And then thirdly, we use the vegetation propagation or cutting. So using this vegetation plot, we can synthesize the mangrove condition or mangrove health, vegetation health. And from the interview survey, we can provide the information about the investment and the effort about the mangrove planting. For example, the project cost, how much the project cost, the propagule, how many propagule planted, and the area of planted by mangroves, and the indication of success. For example, the propagule survival mangrove area increase. And also we can have other information, for example, the planting techniques, nursery techniques, and mangrove propagation. This is supported also by our experiments using cutting. And all of this information together, we synthesize about the mangrove ecosystem restoration. This is important. We can see that what the new approach is required, or just we do the current mangrove planting technology or mangrove planting approach. The result is, we can see here in the map that around 75% of study sites was categorized as degraded mangrove, poor or moderate mangrove. You can see here that all of this area except K Island and Weda Island is degraded mangrove or poor or moderate. And we can, if we go to the detail, we can see here that about 66% of the total 114 research stations. So every site we have some research stations. For example, in here 58 and so on. So 66% of the research station is degraded forest, and only 34% was healthy mangroves. For example, here almost all of the mangrove is degraded. On the K Island, we have healthy mangrove. K Island have 70% of the mangrove is still healthy. And how about the mangrove planting? How much is the money invested in the mangrove planting? You can see here that large amount of money was spent for mangrove plantings, mean that about 12,000, more than 12,000 per locations. And most of the money, 55% spent for planting mangrove, and 37% is for procurement of mangrove seedling or propagules, and land preparations for 80%. No money for spent for maintenance or monitoring. Is very it's not the ideal condition, because mangrove is required for the maintenance and also monitoring. Most of the fund is from the government, 78%, and followed by companies or corporate social responsibility and self-funding in 7%. You can see here that propagule planted is also a high, project cost is also relatively high, and planted area also relatively. And most of the planted area was degraded lands or acts of mining area or ponds, especially shrimp ponds. And 32% was coastal area and 5% was dune area because of the sedimentation. We can see that many effort and a lot money spent for mangrove planting, but you can see here that 79% of mangrove planting was not successful. This is indicated by low seed propagule, about 60%, and mangrove area increase is only 2%, much lower than mangrove decrease. Mangrove area decrease 30%. You can see here that some of the mangrove area is decreased, it's not increased at all. How about the mangrove propagations? Most of the mangrove planted is limited into species. For example, commonly is Rhizophora micronata and Rhizophora apiculata is about 84%, and only 16% is planted by other species such as Gymnorrhiza, Bruguiera gymnorrhiza, Avicennia marina, and Serioptagal as well. And then we can see here that we try to we did experiments using a cutting or vegetative propagation for 10 species of mangrove. 5 species is associate mangrove and 5 species is true mangrove. We can see here that cutting is not successfully applied for true mangroves. No one can survive using a cutting techniques. And associate mangrove is a little bit much better. It's around the survival rate is around 40 to 90%. and the growth rate is 3 to 6 centimeter per month, or mean 5 centimeter months. This associated mangrove is much better using the cutting, but the challenge is how about the true mangrove? This one is the comparison between the current condition and the ideal conditions. Firstly, we have to change our mind for just planting the mangrove, just plant the mangrove. The restoration of the mangrove ecosystem, this is a quite difference, because this one, the mangrove ecosystem focus on the restoration of the ecosystem, not only just planting the mangrove. And currently, most of the mangrove planting, they use a community-based approach. We have to move to the ecosystem and community-based approach by the concept, using the concept of learning from nature, building with nature. Or we also can say that ecosystem and community-based approach, or ICBR, will provide coastal ecosystem community resilience by combining of ecological rehabilitation, biotechnology, local community developments. So this method probably will much better than the conventional one. And for the nursery, until now they still use the conventional nursery and propagation. So we have to change to the biotechnology based nursery and propagation. Why? Because not all of the seeds or propagate are available throughout the year, because it's seasonal. So we have to use the improved nursery, cutting, tissue culture, or biostimulant to improve the mangrove growth. And next is the current condition is without or unsuitable land preparation. Most of them is without or limited land preparation. So we have to change to the suitable preparation. And we have some technology exists. For example, ecological engineering is applied in USA, but we have to test in Indonesia. In Indonesia, we have embankment, kuludan, but this is not also applicable for all area of mangrove restoration in Indonesia. And the new thing is we have to apply the bioremediations to remedy the land, the soil, for example, because just I mentioned before that most of mangrove restoration area is degraded mangrove because of mining or ponds, shrimp ponds. And next one is conventional mangrove plantings. We have to move to or change the improved mangrove planting using, for example, multi-species, natural colonization, or biorevegetation. We use technology to do this one. And lastly, this is the most important because the current condition they without maintenance, almost without maintenance and monitoring. So regular long-term maintenance and monitoring is essential to ensure that mangrove ecosystem restoration is successful. This is including the plant embroidery, pest and disease control system, pollution and distribution control system. And also for monitoring, it is important to monitor the growth and the survival rates, area increase, and also ecosystem. Thank you very much for listening.
So thank you so much. Now we are 3 minutes behind schedule, so we can just move on to the next speaker. So please, Sarwo Handayani, the time is yours.
Good morning. My name is Sarwo Handayani from NIHRD. Thank you for the opportunity to present my research on the title Identification of Malaria Parasite among Macaques in Central and South Kalimantan Province, Indonesia. Currently, malaria is still a high problem in the world as well in Indonesia. Globally, there were more than 200 million cases of malaria in 2019, mostly in Africa, South America, and Southeast Asia. The right maps demonstrate that the endemicity of malaria in Indonesia 2 years ago. Although a lot of provinces and districts had obtained a certification of malaria elimination that represented as a white color, however, some districts, mainly in Papua and East Nusa Tenggara provinces, are still categorized as a high endemic of malaria. Even the malaria cases in Papua is around 90% of all Indonesian malaria cases. WHO states that there are 5 human plasmodium, namely Plasmodium falciparum, Plasmodium vivax, Plasmodium malariae, Plasmodium ovale, and the latest one is Plasmodium knowlesi. In the past, Plasmodium knowledge only infected macaques, but to date, there are many reported regarding with infection of Plasmodium knowledge in human. If you look at the history of Plasmodium knowledge, it started in 1931 when Plasmodium knowledge was isolated firstly from macaque which imported into India. The human cases of Plasmodium knowledge in a large number were reported firstly in Sarawak, Malaysia in 2004, then followed by several countries in Southeast Asia like Thailand, Indonesia, Singapore, Vietnam, and Laos. Outside those countries, the imported cases of Plasmodium knowledge also found in travelers in Europe, America, and also Oceania. Other than Plasmodium knowledge, Plasmodium cynomolgi and Plasmodium inui can infect non-human primates. This fact suggested that those two Plasmodium are potentially cause transmission to human. To date, naturally acquired human Plasmodium cynomolgi infection have been reported in Malaysia and also Cambodia. The main natural host of Plasmodium knowledge is Plasmodium cynomolgi and Plasmodium inui are long-tail macaques, that is Macaca fascicularis, and pigtail macaques, that is Macaca nemistrina. Why Symbiont malaria cases increase? Because the growing of deforestation by the human activities such as road construction or illegal logging and mining that may affect to serious environmental problems such as the destruction of wildlife habitat and also the spread of zoonotic disease, including malaria. Therefore, it is important to determine malaria among macaques to inform the public of the risk of acquiring zoonotic malaria. Previous year, human-infected Plasmodium knowledge cases have been reported in South Kalimantan forest. And similar cases could be taken place in other areas of Kalimantan region, for example, Central Kalimantan, that is similar environmental condition with South Kalimantan. Therefore, the objective of the study is to identify malaria among macaques in two provinces in Kalimantan, Indonesia. Central and South Kalimantan provinces were selected as a location for collecting samples, particularly Palangkaraya and Pulang Pisau district in Central Kalimantan and Banjar and Tanah Laut district in South Kalimantan province. We choose these locations since there are many reports of Plasmodium knowledge in human, and they also have a quite similar ecosystem. The study was conducted in 2014. Sample of the study was caught in peridomestic macaques. The macaques targets were long-tail macaques, that is Macaca fascicularis, and pigtail macaques, that is Macaca nemistrina. However, if we catch another species macaques, they will be included as a sample but not be analyzed. Ethical approval was obtained from Ethical Committee of NIHRD. Around 2 and 3 ml of blood were collected by venous puncture. Then blood be made for thick and thin blood smears and also the dried blood spots. Microscopy and PCR were performed to identify the existing Plasmodium in the sample. This is the workflow of sample collection. Firstly, we prepare the material for collecting the sample. As stated by the local people, we put several bamboo traps with enough food inside. Every 3 or 4 days, we check the trap to see if any macaques have been caught. Since if there are too many macaques in the same cage, they will be fight with each other. The caught macaques then be anesthetized using ketamine. During they were unconscious, we took the blood, examined the physical condition, and also checked the dental structure. Blood smears and blood spots were made for microscopy and PCR examination. As a result, we collected more than 200 macaques from 4 districts dominated by Macaca fascicularis. The proportion of male macaques was higher than female. More than half macaques collection was at young age. The proportion of child and adult macaques were almost similar, but we also found baby macaques in this research. Based on location, the number of macaques collected from 2 provinces was equal. However, macaques collected from Tanah Laut district was the highest, and the contrary was from Banjar district. Macaca nemistrina were found from all districts except from Pulang Pisau. The morphology of parasite can be detected by thin and thick smears with a Ginsa stain. By microscopy, non-human primate parasite can be detected to genus level with a Dalton magnification. However, even the experienced microscopists, it is hardly to define Plasmodium at species level. The below figure illustrates the amplification product from the sample by nested PCR. Plasmodium knowledge as seen in figure A gave an application product size of almost 300 pp, while Plasmodium cynomolgi in figure B gave an application product of more than 100, and Plasmodium innoi in Ficus gave an application product almost 500 pp. This table shows the distribution of malaria parasite by PCR based on the sampling location. We detected almost 50% of macaques infected with Plasmodium. The highest malaria infection was found in macaques from Tanah Laut district, and then followed by Palangkaraya, Pulang Pisau, and Banjar districts. Among the three Plasmodium, Plasmodium innoi is simply dominated over other Plasmodium species. The next table demonstrates the distribution of malaria parasite by PCR based on macaque species. More than half Macaca fascicularis were infected with Plasmodium. And Plasmodium innoi was also prevalent in Macaca fascicularis. Unfortunately, there are some samples have not been identified at the species level, although they are positive of Plasmodium genus. If we look at the type of malaria infection, more than 50% macaques were infected with single infection. Around 15 were infected with multiple infection of 2 or 3 Plasmodium species. Until now, it is difficult to differentiate simian Plasmodium by microscopy only, since the morphology of their parasite almost similar. For example, in the early stage, PK is quite similar with Plasmodium falciparum, while in further stage, it is more like Plasmodium malariae. Therefore, malaria identification is important to define the simian Plasmodium at species level. Plasmodium innoi infection was prevalent in Macaca fascicularis, caught in South Kalimantan province, particularly in Tanah Laut district. It is possibly due to the different distribution of each macaca species and also the existing anopheline factors. The finding of multiple infection in several macaques indicated a highly transmission of malaria in these areas. We still discover and identify Plasmodium species, and the reason could be in the macaques infected by other simian Plasmodium and the limitation of existing primer in our laboratory. There are several published data of Plasmodium in human. Outside Kalimantan, human Plasmodium knowledge was reported in Aceh, Jambi, and North Sumatra. However, to date, there have been no reports of human Plasmodium cynomolgae in Indonesia, although the same cases have been reported in several countries in Asia. In case of Plasmodium innoi, only experimentally occurred human infection has been documented. However, recent public publication reported that the findings of human Plasmodium innoi-like and other non-human Plasmodium infected cases were detected in the indigenous population and various subtribes in Malaysia. In conclusion, the finding of large number of Plasmodium-infected macaques in Kalimantan province increases the potential of cross-species transmission of the simian malaria to humans as a consequence of the close interaction between two primate species. To reduce the potential transmission by zoonotic malaria, an appropriate malaria surveillance and the capacity of health workers in malaria diagnosis should be significantly improved in line with malaria elimination strategy. This is the end of my presentation. Thank you very much for your attention.
So thank you for the nice presentation for all the speakers. Now we can discuss. We can move to this discussion session. So so there's already some question in the chat box. The first question for the Dr. Dwi: if the soil sample was carried out on the pit soil in another location with the same pH, would it reflect the nutrient in the same range, or would the fungal result be close to the location you studied? And also, the same question goes to Dr. Dwi. I was wondering whether mycorrhizal fungi like Glomeromycota group enriched or detected in your metagenomic analysis. If so, is there difference between the two sampling sites? Thank you. So for the Dr. Dwi, maybe you can answer this question.
Okay, thank you to Mrs. Yuni for your question. Okay, I will try to answer your question. According to our research, I also do research by using shotgun metagenomic from soil sample we obtained from South Sumatra with type of non-tidal swampland, and also from South Kalimantan. From other location, we get 5 dominant phylum. the two phylum dominant is the same with this result, and the third, fourth, and fifth dominant is different. So the tendency of two dominant phylum is the same. The two dominant is Ascomycota and also Basidiomycota. But from this result from Sumatra, from South Kalimantan and also Central Kalimantan here, we get three other phylum is Ascomycota, Basidiomycota, Glomeromycota, also Zygomycota. But from tidal and tidal South Sumatra, we get Ascomycota, Basidiomycota, Mucoromycota, Oomycota, and Sopagomycota. This result is also in line with other result from other researcher. He do shotgun metagenomic by NGS from soil sample from Xinjiang, China. I think that's all. And the second question, yeah.
Oh yeah, sorry.
Yeah. Second question for Mr. Muhdar. Mr. Muhdar, thank you for your question. From my research, I also obtained FAM, vesicular arbuscular mycorrhiza, yeah, from phylum Glomeromycota, but just only from South Kalimantan. From South Sumatra, we don't get this genus. Yeah, species we get from South Kalimantan is Acaulosphora colombiana. Yeah, but the difference between community from South Kalimantan and also Central Kalimantan is the diversity we get from fungal microbe in South Kalimantan is more higher, and then Central Kalimantan we also get dominant genus like Aspergillus, also Penicillium, also Talaromyces, Fusarium, Trichoderma, and also genus that's special in acid soil, Acidiella. We can get Acidiella.
Could you make it fast because we are running out of time?
On South Sumatra. That is all. I think that's all.
Okay, okay. Thank you so much for the answer. And now we can go to the next question. It's from Bapak Suyadi, and it goes to Mrs. Senli. Is any microbacteria specifically associated with a certain mangrove species, or all of the species you found are cosmopolitan species? It is. The second question is, please inform us if the microbacteria you found in mangrove ecosystem there have any great potential for restoration, bioprospecting, etc. Thank you.
Okay, hello. Thank you for your question. For the first, yes, all of the species that I found is cosmopolitan species. And the second question is, because my interest in anti-infective, so of course all of the microbacteria that I have found have a bioprospecting, especially for anti-infective. Maybe we have a collaboration in future because we have some interest because in a mangrove ecosystem. Thank you.
Okay, thank you for your answer. I also have a question for you, Dr. Senli. So if I'm not mistaken, you also isolate the microbacteria from mangrove, right? Did you find novel microbacteria from your research?
Yes, but I'm not mentioned in my PowerPoint. Yes.
Okay.
I found it.
And yeah, okay. And as far as I know, that the microbacteria also produce the bioactive compound. Did you also test this compound, for example? I don't know. This is just my curiosity.
Yes. All of the microbacteria that I found in mangrove, all of them I do for the screening for the antimicrobial activity because my interest in this area. So that's why I have done with the 10 different microbial pathogens. So I mean, with this compound that produced from myxobacteria, I done with this experiment, but I'm not mentioned in the PowerPoint.
Okay, yeah. Thank you so much for your explanation. So the next question is for Mr. Suyadi. I don't know whether is he still in here because he write in the chat book that his battery is running out. Otherwise,
Yes, I'm still here.
Okay. Okay.
Using my mobile phone. Sorry.
Yeah. Okay. Thank you. So the first question is, how about your opinion if all of the coastal in Indonesian region to be reintroduction or restoration with mangrove ecosystem? And the second question is, please inform us what are categorized of the healthy mangrove ecosystem, and there are any standard to evaluate these three. Thank you.
Yes, I think the second question is, I think it's good if we can not to restore all of the coastal area, but the degraded or deforested mangrove ecosystem. That is the first one, and then second one, the criteria of mangrove health ecosystem is debatable because, but some of indexes exist. For example, mangrove health index is developed by RCO, the Center for Oceanography, and also from Malaysia we have quality mangrove index as well. But not all of this index represent the ecosystem. So we have to develop the most current index to represent the ecosystem health index. Thank you.
Okay, thank you so much for the answer. Is there any question for everyone, for all the participants?
Yes,
otherwise, I can.
Yeah. Yes, you can see here a lot of mangrove. Here is a lot of variation here in Segara Anakan. You can see, so it's not easy to make to simplify the mangrove ecosystem in the one index. So we have to work together and harder to make it better.
Okay, thank you for your explanation. Yeah, so I think we are running out of the time. So if you still have a question, you can just directly go to the speaker by chat in the chat book. And thank you so much for the fruitful discussion for all speaker and participant. And I hope you enjoy this session. And I give it back to the organizing committee.
Okay, thank you, Dr. Dimas, for moderating the session. Ladies and gentlemen, we will have a break for 10 minutes. So please return to this room at, I think it's around, no, it's at 4 o'clock. Thank you. Welcome back, ladies and gentlemen, to the parallel session 3 in this room. Before we continue to the parallel session 3, let's watch some video session from our sponsor from PT Bioscience Medica.
BioSM. Bioscience Medica Indonesia has been established since 2011 to be the most advanced company in life science research and healthcare technology. We aim to contribute in healthcare sector in Indonesia and increase the society's value through innovation. We dedicate ourselves into bringing impact and outcome in science movement as our core value. We provide reagents, consumables, chemicals, equipment, and services for laboratory needs or related to biotech products that consist of several application categories. BioSM DNA Tech. DNA Tech provides products, instruments, and reagents related to the structure, function, evolution, and mapping of the genome of the branches of molecular biology. BioSM Cell Tech. Cell Tech provides products, instruments, and reagents for the study of the properties of adult and stem cells, both in their basic structure and function in an individual. BioSM Protein Tech. Protein Tech provides products, instruments, and reagents for identifying the processes that cause disease, researching processes in cells, and networking at the protein scale, and studying the properties of protein expression levels, interaction, modification after translation, and others on a large scale. BioSM Diagnostic and Therapy. Diagnostic and Therapy provides instruments and reagents for detection and identification of disease in one condition or to differentiate one disease and condition from others. LabCube is a biosafety level 2 laboratory that can be located anywhere as a place to do COVID-19 testing and other necessary research. Our after-sales services are provided by well-trained personnel such as field application support and field service engineer. We also have regular activities to share scientific insight and help support the development of your research, such as monthly webinar, events, training, visitation, and services. We serve more than 20 provinces in Indonesia through cooperation with resellers. Our market segmentation includes academia, government, industry, and medical. We are looking forward to working together.
Okay, ladies and gentlemen, to lead the panel session 3, I would like to invite Dr. Joko Tribo from Research Center for Biotechnology, Green. Dr. Joko, time is yours.
Thank you. Before I continue, can you hear my voice clearly?
Yes, it's perfect.
Perfect. Thank you for the confirmation. Good afternoon, ladies and gentlemen. Now we are in the last session for today's presentation. In this session, there will be 5 pre-recorded videos from 5 presenters, and each video will be maximum for 10 minutes. So if your video is more than 10 minutes, the committee will shorten your video to 10 minutes. And the first video will be from Sarwo Handayani. So for the committee, if you are ready, please start the video.
Good day, nice to meet you. My name is Sarwo Handayani from NIHRD.
I would like to thank for the opportunity to join this program and have a time to present our research with the title 3 Dominant Genotypes of Human Papillomavirus Among Indonesian Urban Women. Human papillomavirus or HPV is a small double-stranded DNA virus with majority cause of cervical cancer. In the world, cervical cancer has the 4th position in woman cancer after the breast, colorectal, and lung. It also included the 10 highest cancer cases based on global estimation data in 2018. Based on HPV Information Center in 2021, it is estimated more than 36,000 women are diagnosed with cervical cancer, and more than 20,000 die every year. Several studies denote that HPV is not only affect in women, but also trigger another cancer in men. Currently, more than 170 genotypes of HPV have been identified, both a high-risk and low-risk group. High-risk genotype are potential to cause cervical cancer, while low-risk genotype can promote anogenital warts. Among all genotypes, HPV 16 and HPV 18 are the main cause of cervical cancer, with the proportion of 70%. To date, 3 HPV vaccines which commercially available, that is bivalent, tetravalent, and 9-valent. In 2016, NIHRD has conducted a national study of HPV among women in urban areas and collected around 35,000 cervical swabs. After screened, we got 5% were HPV positive. Therefore, the aim of the study was to identify the HPV genotypes among Indonesian urban women. The results carried out in 2018. The samples were archival samples of cervical swabs with the HPV positive and collected in 2016. The criteria of the respondent, age between 25 until 64 years old, living in urban areas, have been married or have had sexual intercourse. And the last one is willing to send the informed consent. The exempted ethical approval was obtained from NIHRD. And the identification of HPV genotype using 2 methods, that is hybridization, which detect 3 genotype, HPV 16, 18, and also 45. And the second one is a PCR hybridization method, which detect more than 30 genotypes. Data then were analyzed descriptively based on respondent characteristics. The result shows that the respondents are mostly at age between 35 and 44 years and passed the senior high school and unemployed or a housewife. Based on a socioeconomic, the proportion of HPV positive was quite similar at all those levels. The left figure represents the hybridization cassette for identification 33 HPV genotype. The positive of HPV genotype are pointed by the purple of color dots. Each cassette can be used for detection 4 samples. 10 most frequent HPV genotype were dominated by high-risk group. If you look at the table, the 3 most common genotype were HPV 52, HPV 16, and HPV 18, with the proportion around 16 to 18%. For the low-risk group, they were dominated by HPV 43 or 44 and HPV 42. These figures illustrate the HPV genotype based on the age and education group. Each HPV genotype, both high-risk and low-risk group, were equally distributed at all age groups and all education levels. The same pattern is observed on these figures. The proportion of HPV genotype were pretty similar among all occupation types and a socioeconomic level. And according to the figure C, HPV infection was dominated by single infection at about 90%, and the remaining infection were caused by multiple infection by 2 or even 5 genotypes. The HPV genotype data in our community is still limited. Based on our research, we found that HPV 52, HPV 18, HPV 18 were dominant, which is similar with the previous study conducted in women aged 15 until 70 years in Jakarta, Tasikmalaya, and Bali. About 4% of women in general population are estimated to get cervical HPV with dominant by 16 and 18. And more than 87% of invasive cervical cancer are attributed to the same genotypes. The dominance of the HPV genotype are varied among countries and regions. HPV 52 genotype was predominantly found in population of women above 30 years old in South China, especially in the Guangzhou area. While in Hong Kong, although it is still a part of South China, but HPV 16 was likely more dominant than others. Our finding revealed that the most infections are caused by single HPV genotype. However, infection with multiple genotypes should be not ignored. HPV 52 is most commonly found together with HPV 16 and HPV 18. Nevertheless, several studies can conclude that the presence of multiple infections is not considered to be directly associated with an increased risk of cancer formation. In conclusion, the three dominant HPV genotypes identified in urban women in Indonesia are HPV 52, HPV 16, and HPV 18, respectively. The last two genotypes have the most potential to cause cervical cancer. Therefore, it is important to screen and identify the genotype of HPV in women, especially in high-risk group. And the discover of the other high risk of HPV genotype in the community should be in the consideration of the prevention efforts such as vaccination using a vaccine that is in accordance with the required high HPV genotypes. This is at the end of my presentation. Thank you very much for your attention.
Sorry, I was muted. Thank you for the presentation. And now we are going to watch the second video from UP Isnaini. Dear committee, whenever you're ready, please start the pre-recorded video.
Thank you for the opportunity. Now I share screen first. Can you look at my slide? Okay. Thank you, ladies and gentlemen. Let me present our manuscript from our study entitled Diversity of Orchid Species in Liwa Botanical Gardens and Their Utilization by the Community. Here, me, myself, UP Isnaini, and our team, Ibu Esti Munawaroh, we are from Research Center for Plant Conservation, National Research and Innovation Agency, and Pak Purwanto from Research Center on Biology, National Research and Innovation Agency. Introduction, Liwa Botanic Gardens, the location is a Pekon Kubu Perahu Village, West Lampung Regency, Lampung. Liwa Botanic Garden has been collected plants from various forest area around Lampung province, such as South Bukit Barisan National Park and the protected forest around West Lampung. At least 6,277 specimens have been collected and more than 3,000 specimens have been planted in various locations in the gardens based on the zoning prepared in the master plan. A total of 123 family, 245 genera, and more than 300 species of plant have been recorded as a plant collection of Liwa Botanic Gardens, including the Orchidaceae or the orchid family. Liwa Botanic Garden is expected to be a reference and a place for conservation and the development of Indonesian ornamental plants, including endemic, rare, and unique orchid species. Orchids, we know, is a high economic value plant family, and at least 880 genera and more than 27 species of orchid have been reported worldwide. And orchids known in the global horticulture market as ornamental plant, or several species of which are classified as edible or scented. And some species have long been used as traditional medicine ingredient. Liwa Botanic Garden Orchidaceae is one of the priority family to conserve in Liwa Botanic Garden. A total of 132 species and 52 genus of orchid have been conserved. And previously, it was reported that 109 specimens consisting of 425 collection number and 60 genera had been collected at the garden. This number is still very limited compared to the diversity of orchid in the world. However, the existence of this collection has been widely used for education and research material. This study aims is to update the latest development of the orchid collection in Liwa Botanic Garden and their use by the community. Material and method. This study was conducted in October 2019 until March 2020. For the first step, data of orchid was collected from Liwa Botanic Gardens registration unit, and a direct check at the net house of orchid based on the orchid collection data from number 1. And this stage, each collection number was confirmed for the existing and existing condition. For the next, we check the name of the orchid species. Verification of the species name was carried out by referring to the plant list website and Plant of the World online. A verified data analysis was carried out using Excel to determine and grouping the number of specimen, genera, and species of orchid collection, as well as genera and species with the highest number of specimens. In addition, literature study was conducted to checking the conservation status, referring the IUCN Red List and CITES website, as well as Indonesian government regulation. The data on utilization of orchid collection by the community was taken from the visit data logbook and literature study. Result in discussion. There is 986 of orchid specimen in Liwa Botanic Garden, including 53 genera, 96 species, and more than 300 specimens of unidentified species. The genera with the highest number of species are Area, about 40 species, Bulbopilum and Celogene 9 species, and Dendrobium 8 species. The highest number of specimens unidentified to species level found in the genera Bulbopilum. There is about 55 specimens. And orchid species with the largest number of specimens is Agrostopilum majus and Bulbopilum lobii with 9 specimens. most of the orchid collection by Liwa Botanical Garden are epiphytic orchid at about 85%, like Celogen, Cymbidium, Dendrobium, Area, and Pomato, Podophyllus, etc. Area is the most widely collected orchid species in Liwa Botanic Garden. And limited terrestrial orchid have concept in Liwa Botanic Garden like Acanthapepium japonica, Arundina graminifolia, Calanthe, Zolingeri, etc. Orchid collection of Liwa Botanic Garden is generally known as attractive ornamental plant, but there is two potential species as medicinal and perfume ingredient like Arundina graminifolia and Dendrobium cruminatum. And from all of this orchid collection, there is two species of orchid that include an IUCN red list like Celogen forstermanii in less concerned category and Papiopedilum tonsum in endangered category. And there is two species of endemic Sumatra like Dendrobium mutigibum and Papiopedilum tonsum. Liwa Botanic Gardens orchid collection has been used by students and public for education, research, and tourism purposes. Here is some picture of the flora education from the junior high school, from the primary school, etc. There is more than 1,000 people, include the students, that visit Liwa Botanic Garden to flora education, and about 64 students has conducted their research in Liwa Botanic Garden. They are from Lampung University and Muhammadiyah Yogyakarta University. The conclusion of our research is Liwa Botanic Garden has 86 orchid specimens consisting of 53 genera, 96 species, and 366 specimens of unidentified 2 species. And Dendrobium has the largest number with a total of 74 specimens. There are 2 species of orchid with protected conservation status in Indonesia, like Grammatophyllum speciosum and Panda sumatrana, and 2 species including in the IUCN red list, namely Celogen forstermanii with the less concerned category and Papiopedilum tonsum in the endangered category. The third, the existence of the orchid collection in Liwa Botanical Garden has been used by the community for education, research, and tourism. Thank you very much for your opportunity.
Thank you. And next, for the next video is from Dini Hari Indah Kusumawati. Sorry. Whenever you're ready, committed, please start the video.
Good afternoon, everyone. Thank you for giving me opportunity to present my study with title Phenol 3-acetic acid of Isobacteria isolated from Imperator cylindrical grassland in Indonesia. Introduction. Land degradation is indicated by invasion of Imperator cylindrical grass, an estimated about 8.5 million hectares in Indonesia. This wealth is well adapted in different soil types, climates, and poor soil conditions. However, it is difficult for croplands to grow in this area because of low soil quality and low soil fertility. In order to improve the soil quality after grown by Imperator grass, indigenous indole-acetic acid-producing bacteria are able to improve the soil fertility. The bacteria improve the lateral root growth and root hairs and improve the absorption of water, ions, and nutrients. The aim of the study are obtaining the diversity and potential of IAA-producing bacteria from Imperata grassland. Materials and method. The bacteria isolate obtained from the previous study were isolated from 4 composite soil samples that grown by Imperata grass, among them SV and RM soil sample from East Nusa Tenggara from different regions, AAP from Katingan Central Kalimantan, CSC from Cibinong, Bogor, West Java. 68 positive isolate for IAA production from the previous study were identified based on the molecular technique. Molecular identification was conducted by colony PCR. The sample were run in PCR machine and then visualized to confirm the quality of the PCR product. The sequence was analyzed by bioedit software, then checked the similarity with the database. All isolate present in 10% of glycerol stock per year, then qualitatively screened for confirmation and quantitatively analysis for AAA production. Quantitative analysis was conducted by growing the bacteria in half-strength TSP and incubated for 0 hours, 24 hours, 48 hours, and 72 hours. Then the solution was added by Salcosi region and read for its absorbance. Result and discussion. Part 1. Molecular identification. The analysis of phylogenetic tree revealed that the 68 IAA-producing bacteria divided into 4 phyla and 14 genera. The phylum of Proteobacteria consists of genus Rhizobium, Pseudomonas, Klebsiella, Pantoea, Parabucolderia, Acinetobacter, Serratia, and Enterobacter. Phylum Firmicutes only consists of genus Bacillus. Phylum Actinobacteria consists of Lawsonia, Mycobacterium, and Arthrobracter. Phylum Bacteroidetes consists of Pedobacter and Chryseobacterium. Here we can see the chart part that show about the diversity of IAA-producing bacteria from Imperata grassland in Indonesia. We can see that the amount of the isolate vary each soil sample. The total amount of isolate from SV soil sample is 21 isolate that consists of Rhizobium, Pseudomonas, Klebsiella, Serratia, Enterobacter, Bacillus, and Pedobacter. 90 isolate from RM soil sample consists of Klebsiella, Acinetobacter, Enterobacter, Bacillus, Lawsonia, and Pedobacter. 14 isolate from KAT soil sample consists of Pseudomonas, Klebsiella, Pantoea, Enterobacter, and Lawsonia. And the last 14 isolate from CSC soil sample consists of Rhizobium, Parabucolderia, Serratia, Enterobacter, Bacillus, and Lawsonia. In this study, we find interesting result of phylogenetic tree from phylum Bacteroidetes. Isolate SV150 was checked for the similarity in ISIB taxon, and the similarity is 98.99% with Chryseobacterium globum. Based from the phylogenetic tree, the bootstrap value of sample SV150 is below than 70%, and this sample is identified as a Chryseobacterium sp. This result indicated that this isolate supposed to be candidate of a new species. However, further study is needed to determine this isolate as a new species. Part 2, qualitative screening and quantitative analysis. In this study, we only 39 IAA producing bacteria. Here we can see that 4 isolates show a pink color that indicated their ability to produce IAA. Here is the table of quantitative analysis result of IAA producing bacteria. 15 isolate from SV soil sample, 8 isolate from RM soil sample, 10 isolate from KAT soil sample, and 6 isolate from CSC soil sample. The highest amount of IAA producing bacteria from isolate from KAT soil sample that achieve 179.57 ppm. 20.5% of IAA producing bacteria were obtained from Klebsiella genus, produce IAA about 115 ppm, with highest amount 179.57 ppm. The previous study reported that Klebsiella isolated from Thailand produced IAA up to 291.97 ppm. Finding of the study showed that our isolate is potential to be developed in the future as a biofertilizer in the land of the grown by Imperata cylindrical grass. The conclusion: finding of the present study highlighted that bacteria isolated from Imperata grassland are capable to produce IAA dependent of tryptophan and consists of 4 phylum with 14 genera. Klebsiella genus achieve highest amount of IAA production than other genera with highest amount 179.57 ppm. Thank you for your attention.
Thank you. And I would like to remind you again about question. Please write your question in the chat and I will read it for you in the end of the video, after the last video. Okay, and we will move to the next video from Sylvia J.R. Lekatompesi. And dear committee, whenever you're ready, please start the video.
Good day, dear ladies and gentlemen. Thanks to moderator. Okay, I will present the result of my research with the title Isolation and Characterization of Extracellular Enzyme from Endophytic Bacteria Isolate from Typhonium flagelliform and Typhonium rumei. And this is our research, our team. This activity is part of the APH DEPA activity, means EPA as the coordinator of the activity to support the National Research Priority Program in the health sector for the provision of standardized herbal medicinal ingredient and phytopharmaceutical. Introduction in this research, the red taro plants in Indonesia is known as the medical plant. The potential of red taro plant is used as an antimicrobial, antioxidant, anti-cancer, and others. What endophytic microbes? What are endophytic microbes? Endophytic microbes are microbes that live in symbiosis in plant tissue. These microbes do not cause plants to die or become diseased. One of study that has been conducted on 8 plants turned out that endophytes have activity that produce enzyme as
amylase, cellulose, and protease, and others. This is interesting apart from the potential of rat taro plants as medical plants. The isolate endophytic microbe may have other potential, not only metabolites that will be used for medicine, but endophytic microbe that produce extracellular enzyme. The role of enzyme in plants, amylase, are synthesized in the fruit during the ripening process so that the fruit become sweeter. Cellulose to form the framework of plant cell walls. Proteases in plants play a role in intracellular and extracellular process such as fruit development and ripening, as proenzyme activity, as food reserves and degradation of damaged proteins. In addition, this enzyme also acts as a decomposer to help plants get simpler compounds to can be used by plants. This aims to isolate endophytic bacteria from rat taro plants and to determine the potential of endophytic bacteria from rat taro plants in producing extracellular enzyme. And the method used, we start from sampling with collaboration with Bangtan to get variant from rat taro plant, then reproduce for research need. And we get 4 variant from the rat taro plants, and we use two in this research. We use two variant, and the number 3, 3, and 4, and this is the taro plants Typhonium flagelliforum and Typhonium plume. And next, we do isolation of endophytic microbes, was carried out using 75% ethanol. And then the sample, we put the isolation media to which nystatin antibiotic was added so that fungal growth was inhibited because our target was only isolate endophytic bacteria from the red taro plants. Next, we do the amylase test. We use starch soluble agar 5%, and we observe the clear zone around bacteria colony. And next, protease test. We use skim milk agar, and we observe the formation of clear zone around the bacteria. Next, test cellulose activity. We use carboxymethyl cellulose, and then we incubate for 48 hours, and Congorate reagent was applied to the surface of the bacteria isolate until the surface was submerged, and then allowed to stand for 25 minutes. We observe the formation of clear zone around the bacteria colonies. And this is the lytic index measure is calculated using the following formula. And the result from isolate of endophytic bacteria from rat plants, we get total 59 isolate. And from the figure, we can see isolation of rat tailor from leaves, and we incubate about 25 hours, and we can see the colony around the leaves. And then we purification of rat tailor endophytic bacteria until single isolate bacteria. From the graph that the number of endophytic is higher in the tuber part of other plants due to the availability of food reserve in this part of plants in the form of carbohydrates or starch. From figure enzyme activity test result, we can see the clear zone around in the colony bacteria in amylase test, and same we can see the test cellulose.
Could you
Cellulose
make it
test.
faster, maybe with the speed of 1.5
Isolate.
or 2?
There were obtained as many as 59 isolate. There were 35 isolate of endophytic bacteria from red tailor plant variety Typhonium flagellus. About 6.9653 millimeters from TF3A4 isolate from red tailor plant. Thank you to Ms. Eva Erdayani, PhD.
Thank you. And we would like to apologize because we cut down the video because we have the limited time, only 10 minutes for the video. And now for the last video from presenter Agnes Natalia Wijaya. Yeah, please start whenever you're ready.
Everyone, this time I will present our research about effect of organic additive on regeneration of orchid hybrid Dendrobium bertachong cross Dendrobium black spider cross Sutignoi. But before that, let me introduce some people related to this research. My name is Agnes Natalia Wijaya, as well your presenter for this research. And then there are Mr. Ida Bagusmade Artadana, Mr. Sulistyo Emantoko Dwi Putra, and the last Mrs. Poppy Hartati Harjo. They are the supervisor for this research. So let me introduce about orchid first. Indonesia is one of the countries with the most orchid diversity. Orchids are ornamental plants and very popular due to their lovely shapes and colors. Dendrobium species are epiphytic sympodial orchids and one of the most popular orchid species, but difficult to germinate in natural conditions and grow slowly. This might give an impact to fulfill on market demand. In order to solve these problems, plant tissue culture or in vitro propagation might be used. Seeds of orchid can be germinated to form protocol-like bodies or PLB and grow into seedlings using in vitro propagation procedures. By adding nutrients to the culture media, in vitro propagation can help in the rapid growth of Dendrobium species under sterile and regulated circumstances. The growth media, plant growth regulator, ambient condition, and other are all important factors to consider when propagating in vitro. There are several distinct types of media that can be used, such as Murasaki and Scoop, Facin and Wen, and others. Salts, vitamins, mineral, carbon source, and growth regulators are commonly found in orchid propagation media. Natural and chemical plant growth regulators are the two types of plant growth regulators. Natural plant growth regulators can be found in organic materials such as coconut water, mung bean sprout extract, and many more. Various effects can be caused by the use of media and plant growth regulators at in vitro plant propagation of orchids. So the aim of this research is determine the composition of media and appropriate organic extract for the fast-growing seeds from cross of Dendrobium bertachong cross Dendrobium black spider cross Sutichnoi hybrid orchids. Next, first method is surface sterilization of explant. Hybrid orchid pots from crossing of Dendrobium bertachong and Dendrobium black spider cross Sutichnoi were harvested and then was under running tap water with detergent, sterilized under laminar airflow cabinet, dipped in 96% alcohol and flame. The cell wall of sterilized pots were split open with sterile surgical blade, and then embryo of hybrid orchid will culture on olive treatment medium. Incubate at temperature around 26 degrees Celsius under a photoperiod 16 hours light. And then subculture hybrid orchid every 4 weeks. For second subculture, add olive treatment medium plus 2% charcoal. And then for the third subculture, add olive treatment medium plus 2% charcoal plus 1 parts per million naphthalene acetic acids. Next, for regenerating cultures, embryo of Dendrobium bertachong cross Dendrobium black spider cross Sutichnoi from pots will culture at Murasaki and Scoop medium or Facin Wen medium. You may select one option, and then with addition of coconut water or mung bean sprout extract, concentration may differ for each treatment, plus 2% of sucrose, and then observe the culture until few weeks. For the result and discussion, we can note that medium and organic compound can give an effect on the growth of embryo resulted from cross of Dendrobium bertachong cross Dendrobium black spider cross Sutichnoi. Based on table 2, different type of medium can result varied response of embryo germination rate, which is Murasaki and Scoop medium giving equal to higher germination rate than Facin and Wen medium, both in full strength or half strength. Its top medium result the higher germination rate of embryo at full strength than half strength medium. Germination of embryo were faster at Murasaki and Scoop medium or Facin and Wen medium with addition of mung bean sprout extract than addition of coconut water. Next, according to table 2, we can know that plantlet which normally and uniformly grow up provide an indication of the right composition medium. The optimum treatment for the observed variable was full strength Murasaki and Scoop medium with 15% mung bean sprout extract and 2% sucrose, which is result 90%, 90%, and 90%, at 4, 8, and 12 weeks respectively. Effect of medium and organic compound to percentage of four-leaf plantlet with many number of roots also observed. The use of Morrison scoop medium result higher percentage of four-leaf plantlet with many roots than Facin and Wen medium, both full strength and half strength. When compared the use of organic compound mung bean sprout extract and coconut water with the same type and strength of medium, the results shown that mung bean sprout extract will produce a higher percentage of four-leaf plantlet with many roots than use coconut water as organic compound. Effect of medium and organic compound to number of leaf and roots also observed. When compared the use of organic compound mung bean sprout extract and coconut water with the same type of medium and strength of medium, the results shown that mung bean sprout extract will produce a higher number of leaf and roots than use coconut water as organic compound. Number of leaf with organic compound coconut water less than use mung bean sprout extract but not significantly, whereas the number of roots with organic compound coconut water less than use mung bean sprout extract because coconut water contains a cytokine which has role to stimulate shoots, not for improve the roots, whereas mung bean sprout extract contains essential amino acids such as tryptophan, which is the most important organic substance in auxin biosynthesis, which can promote root growth, so the plantlet would have many roots. Visually, roots morphology of Dendrobium bertachong cross Dendrobium black spider cross Eutychnoi, which organic compound coconut water usually shorter and less. This happened because high content of cytokine on coconut water. Next, figure 1 shown the pods as result of orchid hybrids, and then for figures 2 shown the development of Dendrobium bertachong cross black spider cross Eutychnoi, orchid seed germination on Morrison scoop medium plus 15% mung bean sprout extract plus 2% suckers. Next, figure 3 shown that treatment A resulted plantlet which have many and long roots than other treatment. For adaptation to ex vitro conditions, selected plantlet must have many and long roots, so they can survive well at ex vitro conditions. Don't let excessive water content at the acclimatization medium to avoid root rot. Another advantage from economic side of using mung bean sprout because mung bean sprout is cheaper than coconut water. At figure 4, shown that selected plantlet had been planted with sphagnum moss on flexible cup for acclimatization stage until the plantlet grow up. So the conclusion is the fastest of embryo germination and growth young plantlet which results from crossing of Dendrobium bertachong cross Dendrobium black spider cross Eutychnoi at composition of medium Morrison scoop medium with addition of 15% mung bean sprout extract and 2% sucrose. This is the end of my presentation. I apologize if there is a wrong word, and thank you for your attention.
Thank you, and please give virtual applause for all of the presenters. And now we are in the Q&A session, and I think because we are in the last session, I hope you are still have a lot of energy. So the question and answer session will be unlimited, right, committee?
Actually, we want to stay until late, but yeah, I think we have around until 5 minutes to 5. 10 minutes, around
Okay.
10 minutes.
10 minutes. Yeah, because we have the limited time also for the Q&A session, and there's only one question in the chat, and I will read it for you. So the question is from Dwi Murti Puspitaningsi, and this question is for Ibu Yupi. So the question is, did you take the photo of the flower of Papiopedilum tonsum in the field? Because the flower in the picture, it's not Papiopedilum. And yeah, to Ibu Yupi Ismaini, please. Time is yours.
Thank you for the, or we call Bu Ita, college. The photo was taken from the nursery of Liwa Botanic Gardens, not original from the field. It's a collection from the Taman Nasional Bukit Barisan, or TNBBS, and the flower is bloom in a nursery. For the name, we asked for the name for the expert, Ibu Irawati, but maybe if it is not clear, maybe Bu Ita can suggest us to the clear name, because we are not the expert of the orchid. Yes, we will very thank you if Ibu Ita can help us to correct or edit our manuscript. Thank you.
Thank you, Bu Yupi. Is there any follow-up question from Bu Ita, maybe?
Maybe when we meet, I can see the picture, because too small to see your picture right now. Okay, it's maybe
you.
like,
Thank you.
Yeah.
We maybe we can take a picture again if there is still another flower. Thank you, Bu Ita.
Yeah. The photo is look like Papilopedilum bulenianum.
Oh, okay.
So you can check
Bulenianum.
It may be because it's not clear.
Okay.
Okay. Thank you, Bu Ita. Yeah, thank you, Bu Ita and Bu Yupi. And now, if you have any other question, you can also ask your question to the presenter. If not, then I think it's my turn to ask the question for the presenters. Please raise your hand if you have any questions. No questions? So then it's my turn. I really like the presentation from all the presenters. It's really interesting for me. And I would like to ask to Ibu Dini Hari about the IAA producer bacteria. So it's interesting to see that the Klebsiella, the genus Klebsiella, produce IAA, but you have a lot of Klebsiella from different fields. And I saw that in the result, the IAA from Klebsiella is really different from one field to another. Could you explain why in the same genus they produce different concentration of IAA? Ibu Dini Hari? Could you hear me? Oh, I think Ibu Dini Hari could not hear the question. Okay, now I will move to the next speaker that I would like to ask is from Ibu Yupi Ismaini about the diversity of orchid again. So you mentioned about two species of orchid that have the medicinal properties, which is Arendina species and also Dendrobium species. Could you explain how the community use these two species of orchid for the medicinal properties?
Okay, thank you, Mr. Joko. For the medicinal plant, I think in Liwa they don't use this, but we find the information from the literature study for the Arundina graminifolia is in China. They use many purposes, yeah, but some of them it will use as antidote. I don't know what is that, diuretic. So many use of this orchid in the reference. But in Liwa Botanic Garden, I think maybe only for the ornamental plant for both of them. Thank you.
Yeah. And next question is for Agnes Natala Wijaya. Can you hear me?
Yes.
Okay, so I would like to ask you about maybe the method or maybe the statistic on how you can sure that the organic medium that you mentioned in the conclusion is the optimum medium for your hybrid plants. Maybe could you explain the method a bit?
Okay, thank you for the question. For the parameters, which is a conclusion that full strength MS and 15% mung bean sprout extract and 2% sucrose is optimum because from the percentage of plantlet which is normally and uniformly grow up, and then percentage of plantlet for leaf with many roots, the fastest day to germinate of the embryo, and then the number of leaf and roots. For the statistical analysis is using ANOVA, analysis of variance one way, and then if there were significant difference, the data will analysis use the Duncan's multiple range test at 5% error level.
Okay.
Thank you.
Okay. Yeah. So do you have any plan for further research about the optimum media? Maybe you want to produce for mass produce, for commercialize?
No.
Do you want to produce your media for commercial purpose?
I don't think.
Yeah?
Yes, maybe.
Okay. yeah, I think it's interesting for the orchid fans.
Yes.
Yeah.
Mm-hmm.
Okay, thank you, Ms. Agnes. And for Ibu Dini Hari, maybe you can give your answer now.
Thank you for question, Mr. Joko. I would like to try to answer your question. From my study, the Klebsiella genus show most highest IAA production than the other genera. So I think that this Klebsiella genus very potential to for production IAA, but I still don't know why this genus show most higher IAA production than others genus. But from the previous study, this Klebsiella show the highest than others. But I have a previous study that show Enterobacter produced more than 2,000 ppm of IAA production. Sorry for this answer, Mr.
No, you don't have to apologize for the answer. And I think it's interesting to follow up the study with maybe to see the metagenome of the species from the Klebsiella to see whether they have difference in the genetics and whether this differentiation of the genetics can also affect the IAA production.
Yes, I think so.
Yeah. Okay, thank you, Ibu Dini Hari, for the answer.
Yeah,
and the last, I have a question for Ibu Sylvia Clecatompesi about the amylase production bacteria. So it's interesting to see how you isolate the endophytic bacteria. Do you have any other method maybe that can gain more of the endophytic bacteria?
Thank you for the question. From the isolate from isolate where activity amylase, we collect more from a tuber plant. Then I only I'm only use the method qualitative, and then we only use the media starch soluble agar 5%, and we like this. We incubate 40 hours, and we observe the clear zone around the bacteria colony like this.
Yeah.
Yeah.
Okay. Yeah. And perhaps if there is any other method for the bacteria isolation, maybe you could mention to us, other than only plating the leaves to the agar, like perhaps the method like dilution.
In test amylase, we use after that we put the iodine we put iodine, and that we can see like that aja, like that the clear zone.
Yeah.
And we can see from the slide the clear zone after we put the iodine. The colony bacteria we can see the clear zone from the colony like that. Only we use only the method qualitative.
Yeah. Thank you for the answer, Ibu Sylvia.
Okay.
And now is the end of the session. So I think this is the end of my time to be the moderator in the session, and I would like to apologize if I have the error in guiding
I'm
this
sorry,
session.
Pak Joko.
Yeah.
I'm sorry, Pak Joko.
Silakan,
You don't
Ibu.
have any question to me? You don't have any question to me because every presenter have a minimum one question. Please.
Yeah, actually, I have the question, Ibu, but I think if I allowed by the committee, may I have the one question for Ibu Sarwo?
Actually, we run out of time now, Dr. Joko and Bu Sarwa.
Yeah, I apologize, Ibu.
Okay, thank you.
Okay, so now I would like to give back the time to the committee.
Okay. So thank you very much, Dr. Joko Triwibowo, for moderating this session. And Bapak Ibu, ladies and gentlemen, before we close the session, please kindly open or activate your video because we want to take further session. So Bapak Ibu, please activate your camera, activate your video since we are taking picture together. Okay, I think we start now. 1, 2, 3, and then for the second slide, 1, 2, 3. Okay, thank you very much, Bapak Ibu. And before we close our session today, let's return to the main room for some announcement for tomorrow's agenda. Thank you very much.
Recording in progress.
Ladies and gentlemen, welcome back to the main room of ISIBIO 2021. We would like to thank all the speakers and moderator for the insightful session. Now we have come to the end of the first day of the 8th International Symposium of Innovative Bioproduction Indonesia on Biotechnology and Bioengineering, ISIBIO 2021. We would like to remind you for the second day of ISIBIO tomorrow, we will open the registration at 7:30 and start at 8 a.m. Jakarta time. And for tomorrow, we will have keynote speaker Professor Duk Jo from Asian Federation on Biotechnology, and plenary session we have speakers Professor Zihua Jiang from Department of Animal Sciences, Washington State of University, USA, and Mr. Zahir Ahli, PhD, from Center for Desert Agriculture, King Abdullah University of Science and Technology, Saudi Arabia. And at the end of the symposium tomorrow, there will be announcement of 6 best presenter in ISIBIO 2021. So stay tuned, and we hope to see you too tomorrow. We would like to thank all of you for your participation, and on behalf of the official committee, we would like to apologize for any inconvenient matters during this today event. So see you tomorrow, and goodbye.